Good afternoon. If you are in the United States, and good evening if you are in Europe, or in Brazil, in Mexico, some other places in a different time zone. Before we start here, let me give a thank you to all healthcare workers throughout the world that keep us safe throughout this crisis. And without much further ado, let me turn the floor to the Chancellor of the University of California, Davis, Chancellor Gary May.
Thank you, Walter. On behalf of UC Davis, I want to welcome everyone to our second COVID-19 Public Awareness Symposium. It was encouraging to see such a tremendous response to the first symposium. More than 1,000 people logged in from throughout the United States and Canada, as well as Germany, Brazil, France, and India. As the COVID-19 pandemic continues, we're all looking for answers about potential breakthroughs, ongoing health risks, how our hospitals are prepared, and so much more. UC Davis is determined to be part of the COVID-19 solution and serve the public good. This symposium represents those efforts. Now I have the privilege of introducing a very special guest, UC President Janet Napolitano. President Napolitano has been a strong and effective advocate for the UC system since becoming our president in 2013. This is a tremendous undertaking. The UC system includes 10 campuses, 5 medical centers, 3 affiliated national laboratories, and a statewide agriculture and natural resources program. The UC system has more than 285,000 students, 227,000 faculty, staff, and an annual operating budget of $39.8 billion, and more than 2 million living alumni. President Napolitano is the first woman to serve in this role. She has proven to be a forceful advocate for hardworking students and work diligently to make sure our students have access to a world-class UC education. President Napolitano is a distinguished public servant who has served as U.S. Secretary of the Department of Homeland Security from 2009 to 2013. She was also a 2-term governor of the state of Arizona. Thank you for being here today, President Napolitano. Please join me now in welcoming her.
Well, thank you, Chancellor May, and good afternoon to all. It's an honor to be with you today, albeit virtually, in the company of so many people on the front lines of responding to the COVID-19 pandemic. You know, hard times tend to reveal an institution's true character. From where I sit, I'm actually at my dining room table, but from where I sit, nothing could be more clear. This moment in history has demonstrated what the University of California is all about. This is an exceptional institution whose power is multiplied by our 10 campuses, not to mention our medical centers, our national laboratories, our division for A&R. I am so impressed by the dedicated people I meet at every corner of the university who are engaging in such heroic efforts to help the world face and ultimately triumph over this new virus. I want to especially acknowledge the leadership of Chancellor May. We are indeed lucky to have him as our Davis Chancellor at this time. And I also want to especially thank UC Davis and Dr. Leal for convening this symposium. It is incredible what can be accomplished when you get great thinkers together to listen to each other, to collaborate, to innovate, and to make possible creative new solutions. UC Davis was amongst the first hospitals to face COVID-19 impacts directly. And the world is witnessing the grace and skill with which the people of UC Davis and UC Davis Medical Center continue to meet this challenge. So today, I hope and believe that this symposium will continue to spark new conversations, ideas, projects, and partnerships. You know, when the going gets tough, one of UC's great strengths is in driving collaboration. Faculty, students, and staff alike come together and address problems using all available minds and methods. Indeed, we need all the brain power and talent on deck to get through what we face today. And we have got such incredible brain power and talent at the University of California. This is an impressive community of thinkers and doers. That includes researchers, medical professionals, educators, and others who also happen to be undocumented. So the intersection of these two issues is something I'd like to address today, because even before the COVID-19 pandemic, many of us at UC have recognized that undocumented students, faculty, and staff are such an important part of the UC community. That's why in the year 2017, we sued the Trump administration to stop their attempted rescission of the Deferred Action for Childhood Arrivals program, also known as DACA. DACA was a program that was developed while I was Secretary of Homeland Security and was designed to protect young people who came to this country as children, typically under the age of 6, who have grown up in the United States, gone to school in the United States, done everything they need to be admitted to the University of California in the United States. They really only know the United States as home. They're American in every way, except that they are undocumented. Congress proved repeatedly unsuccessful in protecting this group from the fear of deportation. And so DACA was developed to defer any deportation for young people who applied, who passed a background check. And it also, once they got DACA status, enabled them to work. So that if, for example, they were a UC student, they could work to help pay for their college education. So we have been in federal courts since 2017. In that year, we did obtain an injunction against the rescission of DACA. That enabled the program to continue and for young people to keep re-enrolling and keeping their DACA status. It went to the Court of Appeals. The Court of Appeals affirmed that injunction. There were 2 other cases filed in other parts of the country. They also received injunctions. So the point of fact is that of the many federal courts that have reviewed the rescission of DACA, all have ruled that the Trump administration acted illegally in violation of federal law in how they rescinded the program. They enjoined the rescission, allowed DACA to continue. But then we went to the US Supreme Court. The case was argued in early November. We expect an opinion any day now. All of us get up early to be on the website at 7 a.m. Pacific time, because the Supreme Court posts their opinions at 10 o'clock in the morning. And we are waiting for them to rule on whether the Trump administration properly rescinded DACA. During the pendency of the case, of course, the pandemic occurred, and supplemental briefs were filed demonstrating that some 27,000 DACA recipients are also healthcare workers on the front lines. Some are nurses, some are respiratory therapists, some are physicians, to subject them to the fear of deportation at the moment of this huge public health crisis just seems doubly wrong at all levels. So we need them, we need their resources and their talents, as well as those of all the DACA recipients. There are some 800,000 DACA recipients now in the United States. So we will continue to fight for our DACA recipients, and indeed for all the undocumented recommended members of the university community. Before I close, I want to give special thanks to the healthcare workers and other frontline staff who are doing so much and taking personal risks in their own right to take care of patients and to keep vital functions of our campuses and our hospitals going. And I want to thank all of you for your dedication and your hard work on behalf of the University of California. So I wish you a productive and successful symposium. May you and your families stay healthy and stay safe. Thank you very much. And now back to Dr. Leal.
Thank you very much, President Napolitano. Thank you very much, Chancellor Gary May. So now we continue, and I have the pleasure to introduce the first panelist of the day, Dr. David Labraccio. He's a Vice Chancellor of Human Health Science and CEO of the UC Davis Health System. Dr. Labraccio has committed the organization to expanding care for the unserved while creating a sustainable economic model, earning UC Davis Health public acknowledgement as a leader in caring for medical patients. Dr. Labraccio has repositioned the system to advance healthcare and science into the future and with a nation-leading models of teaching, team-based care, whole person wellness, and innovation programs to harness technology while maintain a patient-centric ethic of care. Dr. David Labraccio, I'm going to stop sharing here, and you have the floor.
Well, thank you very much. It's truly great to be here and have an opportunity to address so many of my colleagues here. We're going to talk briefly about the role of UC Davis in its diagnosis of the first community case here in the United States. Many of you know probably that at the first, the United States felt that all the cases of COVID-19 would be appearing from offshore. And we ended up with the transfer of a very sick patient who our astute clinicians very quickly realized could be a COVID-19 patient, and indeed was diagnosed eventually after spending almost a week in our hospital with COVID-19 as the first community spread. And during the course of that patient's treatment, we discovered a lot of things, confirmed a lot of things that, again, are even perhaps still being debated, which is that the primary mode of transmission of the disease is through droplet spread and is not airborne. That remdesivir treatment, originally actually studied interestingly at UC Davis's veterinary school in terms of its efficacy on other diseases, was applied in a compassionate use and resulted in an excellent result. And that the way to diminish complications with patients as sick as our patient was on a ventilator using prone positioning protocols. So again, being this was the first community acquired case, the fact that our clinicians on the front lines did such an amazing job taking care of this one patient really moved things forward in terms of medical knowledge, but it also moved things forward on the national dialogue about what should our testing algorithms be, to whom should we offer tests, and what these tests might or might not tell us about community spread. I know there's some questions out there about that. And it also signifies the amazing place that academic medical centers continue to have in the fabric of American healthcare. Without academic medical centers, this level of accumulation of subspecialists wouldn't be possible that allowed a diagnosis basically out of thin air. And without academic medical centers, there would not be a race to a cure or a race to the creation of a vaccine, both of which I think you'll hear more about from Dean Allison Brashear from our School of Medicine later on. And because of this, and because of the ability we have to talk about this in our community, I think that's one of the reasons that Sacramento ended up with one of the lowest, not one of the lowest rate of any major metropolitan area of COVID-19. And that's because the presence of this first case actually, I think, made the whole community sit up and take notice as we were in the news and say, wow, it really is here. And we really should pay attention to the physical distancing, the aggressive hand hygiene, and other disease avoidance things such as sheltering in place. And so we've had a tremendously great citizenry out there to whom we need to give great thanks for not spreading the disease in our community. But even today, there are many things we don't know that I think UC Davis is uniquely positioned to study and hope to encourage people to think about this. We have one of only family caregiving institutes that studies how to take care of your loved ones at home. And there are not very many other organizations. This is located actually in our School of Nursing. And they're not really studying this, and yet the familial transmission via continued close contact is one of the major modes of transmission of this disease. And we need to be devoting a lot more resources to helping caregivers all across the nation and the world not end up sick themselves. The other thing that a lot of people have asked and that we're studying and that we don't yet have the answer to is, yes, we know that coronavirus can be detected after people share their germs on handrails and cardboard boxes and money and whatever, but we don't know if those virus particles are viable, can actually cause transmission of disease, and in essence are able to actually infect other people. So studying that and getting a better idea of what we need to be scared of, and frankly, what we don't need to be scared of, is really critical to going back to enjoying our life and avoiding both contamination and potential transmission of disease. And it is just the symposia like this that are so critical to refining our quest for truth and answers, because I'm sure there's of the, I don't know, hundreds and hundreds of people already on the chat here. It's amazing, actually, how many great ideas are out there to further refine what we need to know in order to pursue life productively while we seek both a cure and a vaccine. And last but not least, I want to touch on the inequities of COVID-19. UC Davis Health, and UC Davis in particular, and UC writ large, is amazingly committed to equity and to reducing disparities, both in education, but also specifically in healthcare and the outcomes of healthcare. And I must say we are failing miserably, at least so far, because the overwhelming amount of mortality in our African-American communities should bother us greatly. We don't know why. We don't know, is it a particular type of coexisting morbidity? Is it the lack of access to excellent healthcare and testing at an early stage? Is it diet? Is it whatever it may be? Our underserved communities are suffering disproportionately. We need to study it, and we need more to intervene on it. I'm happy and proud to say that UC Davis Health is partnering with the County of Sacramento. We are doing surveillance testing in some of our most underserved communities to understand if it's more disease or is it just more severe disease that's leading to these disproportionate results. And we're also going out and doing point-in-time testing at whole facilities where we've congregated people at high risk, such as elder living facilities. We need to intervene aggressively and not just simply accept the fact that nursing homes could be wildly infected, and we're only seeing the tip of the iceberg. So we are doing as much as we can possibly do to reduce the inequities in care, and we will continue to do so because that is our role as the University of California, is to better the healthcare of all Californians and of people around the world. With no further ado, I'm going to turn it back over to Dr. Leal, and thank you so much for letting me participate.
Thank you very much. So we shall continue now. And the next speaker and next panelist comes from San Diego, Dr. Atul Malhotra. Dr. Malhotra is a research chief in pulmonary critical care and sleep medicine at UC San Diego. He's Peter C. Farrell Presidential Chair and Professor in Respiratory Medicine at UC San Diego. He has published as many as 325 original papers and plus about 250 reviews and chapters on the topic of respiratory disease since coming on the faculty in the year 2000. Dr. Malhotra has been involved in COVID-related research, including a leadership role on the tocilizumab, that's an antibody that has been utilized for the study for COVID pneumonia. Dr. Malhotra, let you share the slides now. Thank you for your participation.
Thank you. Can you hear me?
Yes. Excellent.
Well, thank you very much. It's my pleasure to be here. I'd like to thank the organizers for inviting me, and it's obviously a privilege to share the podium with such distinguished speakers. I'm going to give about 5 minutes on COVID ARDS. ARDS stands for Acute Respiratory Distress Syndrome. It's a severe form of respiratory failure, which is what we're seeing currently in the intensive care units for patients with severe COVID pneumonia. By way of background, there's sort of two major types of clinical presentation with COVID. There's sort of milder disease where patients either have no symptoms or very mild symptoms, maybe a runny nose and some fever and whatnot. Then there's more severe disease with critical illness, respiratory failure, shock, what's called multi-organ failure. And that tends to be fewer cases, maybe 5% of them, but these numbers are heavily predicated on how aggressively you're testing. If you only test the most severe cases, then clearly the numbers look worse as far as the percentage of patients who are doing poorly. If you test a lot of asymptomatic people, then more people fall into that mild category. You hear a lot of people on television and other places saying this is not ARDS, it's not the acute respiratory distress syndrome, it's something else. And one thing that's interesting as we see these patients in the ICU is how variable it is. So there's a Berlin definition of ARDS that was published in the Journal of American Medical Association. I can tell you almost all the patients I see do meet the Berlin definition of ARDS. And so I think that that phrase is probably not accurate. But that said, it's very interesting to talk to the people in New York who saw a lot of cases, and each different hospital there has a slightly different take on what they're seeing. Some say there's a lot of blood clots in the lung and they're giving heparin, an anticoagulant, and thrombolytics. Others are seeing a lot of mucus hypersecretion and they're doing bronchoscopies and bronching these patients to remove the mucus in some cases. There's some evidence in cases of capillary leak where the lung is flooding with fluid. Other people are seeing alveolar flooding from high surface tension, maybe the surfactant in the lung that's supposed to keep the lung nice and compliant is messed up. Other people saying it's more of a cardiac disease from heart failure. And so there's all kinds of different impressions. And it's usually correct that the right answer is all of the above in the sense that it is slightly different in different patients. But we're seeing a lot of this capillary leak and flooding that I alluded to. What do we do about this? The bottom line take-home message is that good critical care is here to stay. And we've done work on low tidal volume mechanical ventilation. The ARDS network has determined that the mechanical ventilator can be life-saving when set appropriately, but it can also be life-threatening if set incorrectly. So the ARDS network did a study looking at a certain tidal volume delivered by the ventilator to help the patient breathe. And there was a big mortality difference depending on what the settings were on the ventilator. The lower tidal volumes were better, meaning if you give big tidal volumes and you stretch the lung, you can damage the lung and create a problem. So the goal is to do no mechanical harm with the ventilator, and so we set the ventilator accordingly. And despite all the magic potions and other things you hear about, at the end of the day, it's good critical care that is helping these patients more than some of these medications that may or may not work. There's also some talk that some of the lung injury here may be self-inflicted. The traditional teaching is you can't pop your own lungs. If you try really hard to breathe with a closed airway, you're not going to pop the lung. But there are evidence of high shear forces and something called stress amplification that can happen. Some of these patients are breathing spontaneously very aggressively. That is, they take in really deep breaths, and that can actually create a distending force in the lung that can be injurious. That is to say, the lung can be injured by aggressive breathing patterns. There's some experimental data in animals that have suggested exactly that. So there's a school of thought saying the very aggressive breathing or the very deep breathing that's sometimes seen in these patients may actually be injuring the lung at some level. What to do about that exactly is unclear, but it's a source of ongoing research. I wanted to say a little bit about tocilizumab because I was asked to speak about that. There's a company called Genentech and Roche that have a study. It's also partially funded by BARDA, which is a government group through Health and Human Services that's funding sepsis. I can tell you the study is ongoing. We're hoping to complete enrollment next week. Currently, we don't know if this works or not, and so we should complete enrollment within the next week or so, and we should have a result soon. If you look at the existing data, though, the C-reactive protein, one of the markers of inflammation, does seem to improve with tocilizumab. The body temperature seems to improve. The amount of oxygen required seems to improve. And the oxygen levels, the oxygen saturation seems to improve. If you do a CAT scan, there's some evidence of improvement there as well. But we need large-scale, well-controlled studies before we draw any conclusions. So the pictures look pretty, but we need really rigorous data before we can really advocate this type of treatment. Tocilizumab is also known as Actemra. It's that drug has been used in other cases for things like cytokine storm and other conditions. But for now, we're trying it with COVID pneumonia, and we should have an answer soon as to whether it works or not. There is also the question about why do we need these randomized trials? Why don't we just give treatment to people and see what happens? You hear people on television saying, well, what do you have to lose? Just try it. And there's several problems with that approach. The first is we'll never learn the truth if we just give treatments sort of haphazardly. If you just say, let's give this treatment because I think it might work, we don't know if it does or not. Some patients will get better, some will get worse, and we have no way of knowing what's working. All medications have risk and benefit and side effects as well as cost. And so if you just start giving medications that may have risk to them, hydroxychloroquine is a good example where there's potential risk to that. Now people have warned against it. The other issue with medicines like hydroxychloroquine, though, is we'll deplete the supply of these medications for patients who actually need them. So we know hydroxychloroquine works well in patients with rheumatoid arthritis, with systemic lupus erythematosus. It also works well in malaria conditions that are caused by plasmodium. And malaria kills more than 500,000 people per year. And right now there's pretty much no chloroquine or hydroxychloroquine around. That's going to be a problem as the world's supplies have been depleted because they're being used in COVID cases when we don't even know if it works or not. So far, the data are not terribly convincing. I will also say that recovery is a big part of what we're looking at as well. So at University of California, San Diego, we have an ICU recovery clinic that's led by Amy Bellinghausen here. And she's looking at something called post-ICU syndrome and looking at anxiety, depression, post-traumatic stress disorder. We used to sort of declare victory if a patient lived after a stay in the intensive care unit, but now we're hearing more of the stories and we're testing their brain function, their function after critical illness. It's an important part of full recovery after critical illness. And so this is something we have a lot of pride in terms of following patients until they're back to square one. So I'll conclude by saying severe respiratory failure from COVID needs good critical care. There's some reason for optimism. We've heard about remdesivir in the news. There does look to be some promising data there. We should have some more data from ongoing randomized trials soon. We certainly need more randomized trials and good rigorous research to draw conclusions. And I always say, stay calm and wash your hands. This is not Ebola virus. It's not something else. We'll get through this. The death rates will be slightly higher than influenza, but it's not going to wipe out the human race or any of these other dire predictions. So just stay calm and wash your hands. I'd like to thank you all for your attention and thank you to the moderator.
Thank you very much. Let me share from this side here. Very inspirational introductory remarks. It's going to generate lots of questions, I'm sure. But before we get to this kind of questions and start to answer them, let me introduce the next panelist, Dr. Monroe Mavericks. He was here also in the first COVID symposium, and he's back now. He's a professor of dermatology and immunology at the UC Davis Medical School. He's a leading physician scientist with experience in conducting cutting-edge basic science and clinical research. He's the recipient of the Presidential Early Career Award for Scientists and Engineers, also a recipient of the NIH's Director's New Innovator, and a fellow of the California Academy of Science. Dr. Mavericks, thank you very much for coming, and let me send the floor to you, and now you can share your slides.
Thank you, Walter. Of course, slight technical difficulty. Let me see what's. Oh, here we go. Okay, so I want to just briefly recap some of the things we talked about last time, just in case you weren't here. We criticized the CDC hand hygiene policy. Their policy was clean your. I know Dr. Atul just told us to wash our hands often, which is true. But the CDC said clean your hands often, wet, lather, scrub for 20 seconds, rinse and dry. And to only use an alcohol-based hand sanitizer if soap and water is not readily available. And then they say don't touch your face. So we criticize just about every component of this hand hygiene policy. We believe that an effective hand hygiene policy should be feasible, tolerable, and the frequency should be maximized. So our hands are not designed for frequent hand washing. Hand washing changes the skin barrier. About 15% of the US population has a genetic defect in their skin barrier and they cannot excessively wash their hands. Even individuals with a normal skin barrier could only wash their hands about 10 times a day if they're really gonna do that 20-second scrub. The CDC is emphasizing this 20-second scrub. So we talked about the more important thing is to maximize the frequency of implementation. So we want you to practice good hand hygiene as frequently as possible. In order to do that, we need to recognize that you have a certain intrinsic hand washing tolerance. That means that each individual will be able to wash their hands a different number of times a day, depending on their genetic makeup. We emphasize on washing hands after contact with contaminated surfaces or when your hands are soiled. So of course, if you go into a shopping center and it looks like you might have come in contact with some kind of contaminated surface, it's very important to wash your hands at that time. But as important as hand washing is the use of alcohol-based hand sanitizers as frequently as possible, because this way you could increase the frequency of practicing good hand hygiene. We recommended a wearable hand sanitizer. That's a hand sanitizer on the outside of your garment. You don't need to reach into your purse and contaminate other surfaces when you want to sanitize your hand if the hand sanitizer is on the outside of your garment. Wearing your hand sanitizer on the outside of your garment will be a constant reminder of using it. And again, the goal is to sanitize your hands frequently throughout the day, not just do this 20-second scrub, which we could only tolerate about 10 times. Okay, we also talked about the importance of wearing masks. So today we're going to talk about survival and what you don't know. But before we talk about what you don't know, I just want to reemphasize what you probably should know by now. If you're attending this COVID webinar, you're probably well educated, but I definitely want to cover the points that are already out in the public and everybody should know. The first point is that the chance of getting severe COVID disease increases with age. So if you're over 85, you have a really high chance of getting severe disease. And if you're young, chances of you getting severe disease is very low. And these numbers are going to change dramatically because they're dependent upon how frequent the testing is done and how if people who are barely symptomatic are getting tested. But basically right now, if you get COVID at the maximum maximum or minimum, depending on how you think about it, about 72% of the patients are going to have mildish disease that they don't need to go to the hospital. 14% of the patients are going to go to the hospital but not get admitted to the ICU, and about 6% are going to be ICU admissions. But again, these numbers are probably overestimates. But once you get one or two major conditions like heart disease, lung disease, kidney disease, asthma, diabetes, or obesity, these numbers are obviously going to go up pretty high as 27% of the people are going to need to go to the hospital and be admitted, and 13 to 16% are going to get ICU admissions. And if you don't have any real major medical issues and you're a young person, very few people are going to be going to the ICU. Now, that's what you should know, and if you don't know, now you know. But one important thing that I feel your inoculum, how much of the virus you're going to get inoculated with. And this is one of the reasons why good hand hygiene is very important, because although this virus might be widely in the community and we might not prevent contact with it, be able to prevent total contact with it, at least we should be able to lower the viral load that we're coming in contact with. So from animal data, we know that viral load at the time of inoculum, at the time you get exposed to the virus, is very important to determining how severe you're going to get, how severe disease you're going to get. So this is some animal studies with MERS, which is a coronavirus infection. It's a Middle East respiratory syndrome. This was a big thing before the SARS outbreak. It shows that mice that are infected with MERS, if you could decrease the inoculum dose by 50%, you could basically improve the survival by 100%. So it's very important that if we do get inoculated, we try to do things like good hand hygiene, social distancing, because those things will decrease the amount of virus we're exposed to. And the amount of virus, if we base it upon animal studies, should directly relate to our survival and also our ability to get infected. So these are mice, again, getting infected with MERS. Even the mice that got low infection with MERS, they end up having good protection to rechallenge. So these animals don't seem to have any disadvantage of getting infected with a low viral burden, because they're still able to protect themselves if the investigator comes back and tries to rechallenge them. But it's even more bizarre than just viral load at time of inoculation. It's also the volume. There's some studies in animals that the more volume you give, the more viral particles the animals will end up being infected with. So if you use the same amount of virus and you infect animals with different volumes, then you dilute that virus in different volumes, then the amount of the severity of infection is going to be directly related to the amount of volume that they get. So this would also be a recommendation for wearing a mask, because even if you do get sick, hopefully the mask filtered out some of those droplets, and the amount that you get sick with is going to be less. And we're talking about small differences make a big difference in terms of outcome, at least in the animal setting. And it also is age dependent. This is an experiment with influenza virus. They're looking at the amount of droplets that are used to infect the animals. The animals are infected with the same amount of virus, but in different volumes. And it turns out that they're infected with a small volume, the older animals do pretty well, not quite as good as the younger animals. But as they get infected with larger volumes of virus, even though that there's the same number of viral particles in there, you could see that the animals do much worse. We have well, let's go to the next slide. So what about the human data? Can we tell from the human data, are these animal experiments relevant to any of the human data that we're seeing? And of course, these experiments are impossible to do in humans because we can't infect humans with different amounts of virus. And there's no way of knowing really for sure how much of that inoculation was when that patient got sick. But if we look at patients early on when they present, before they're on any type of treatment and early during their disease, we could see that there's a direct correlation between the amount of virus that they have in their respiratory system and how severe they're going to be. So this is survival, and at low amounts of virus, people survive very well. And as the viral titers get higher, this is done by real-time PCR, the worse the patients do. And you get up to like a 55% death rate with SARS. This is pre-COVID-19, but the original coronavirus outbreak, the SARS-1, I guess you could call it, people with high viral titers did very, very poorly with that virus. So I'm assuming the same thing would be with COVID-19. And if we look at MERS, which is also another coronavirus infection, basically the same results were found. If patients are alive, they usually had lower viral titers, and the patients who died had much higher viral titers. patients who went into the ICU had higher viral titers versus the patients who went to the ward. And the patients who had severe and not severe disease, it was based upon their viral titers. So with that said, I encourage you to do good hand hygiene and to wear masks, because if you, in theory, if you do catch this virus, but you are inoculated with just a small amount of virus, hopefully you won't have such severe disease. That's all I want to talk to you about today.
Thank you very much, Dr. Mavericks. So let's continue here. Let me share again. And we are going to move to the next panelist that I'm going to introduce now, Dr. Bill Eisenberg. Dr. Bill Eisenberg is Sutter Health Vice President and Chief Quality and Safety Officer. Sutter has 26 campuses, 5 medical foundations, 65,000 staff and clinicians, serving 3 million people within more than 100 communities in Northern California. Dr. Eisenberg, he is an OBGYN by training. He oversees the overall quality of care delivered across the continuum, as well as patient and staff safety. He's serving his second term on the Hospital Quality Institute's Board of Directors, the quality arm of CHA. He's also co-chair of the Bay Area Council's Healthcare Community. Dr. Bill Eisenberg, thank you so very much for making the time to be here today. You may now share your slides.
Great. So thank you very much, Dr. Leal, and I really appreciate the opportunity to speak with you all today. I thought that it might be well for the audience to understand how our healthcare system, like virtually every other in the state of California, mounted its preparatory responses and some of the lessons we've learned as we've gone through this COVID crisis with all of the rest of you. So without further ado, let's move on. We at Sutter Health, probably like all other organizations in California, were aware of an outbreak of virus that was reported in our emerging infectious disease literature in early January. In fact, I remember sending out our first announcement in January 9th of 2020. Based on what we were seeing in the rapidity with the appearance of cases, we decided on the 23rd of January that we would activate our emergency management system incident command. This is a process we do whenever there are things like wildfires. We've activated them in wildfires, in floodings, a lot of emergency situations, but this was a quite different one because it really touched our entire healthcare system, unlike wildfire, which may just affect areas up near hospitals that are really near the foresting areas. With the activation, we expanded and deployed many, many services that we don't usually provide, but we wanted to be sure that we were ready. So for example, we erected surge tents at most of our hospitals. The idea here is that we can keep patients who may be coming in separated in our emergency rooms from those who have other emergencies that need care, but we did not want to expose them to potential respiratory infectious patients. In addition, we set up drive-through remote clinics so that we could test patients with respiratory symptoms, again, without exposing patients in our ambulatory settings to such potential illness. These drive-through testing sites made it very convenient for patients and also avoided them having to wait in waiting rooms, potentially ill themselves. Lastly, and probably most importantly, we, like I know just from conversations with Dr. Labraccio, we developed our own surge plans to essentially triple the amount of critical care capacity in case we needed it. What that means when I talk about tripling our capacity, that means we increased our 283 critical care beds across the system to be able to care for 900 patients in critical care conditions. So this was a vast upregulation, if you will. And to do that, to be able to, it's one thing to have the beds and the ventilators to manage patients like that, but it also meant that we retrained nursing staff who in other situations may be working in our ambulatory surgery centers so that their competencies were sufficient to oversee ventilated patients. We also enrolled volunteer physicians across our network, many of whom normally work in the ambulatory setting, to be retrained so that they could care for patients in the hospital should the need arise. And then, like many other hospitals, any surgical procedure that was non-essential, non-emergent, we strongly recommended that our patients defer those procedures until such time as we were confident we could render safe care and not need those beds for other reasons. So what was one of the most important lessons we learned? Well, first of all, telehealth certainly was on the rise during this, and it's definitely something that I think we can all predict is here to stay. We had been working over the last several years to lay a foundation so that we could start providing a lot of telehealth services. And our people that were thinking about this on the IS side, our clinicians who were embracing it, we predicted that it would take about 18 months for us to get this going for 14,000 providers across 26 hospitals and about 1,000 ambulatory settings. Part of this was just due to the fact that there were difficult barriers regarding the charging, the privacy and security over the web. These kinds of procedural steps made it somewhat challenging. Suddenly COVID comes along, and this crisis, if we can think of any silver lining, this is certainly one of them. We had a lot of relief and waivers regarding telehealth services rendering. And what did we see? Well, I think these numbers tell us everything that we need to know. We increased our video visits from 15 to 20 in a day to 6,400 in a day by the end of April. So this, you can see that's a 32,000% increase in the number of visits we were doing. We had 50 clinicians who had been formally trained in how to do telehealth visits. And by the end of April, we had almost 4,700, another almost 9,500% increase. And you can see the number of minutes. We went from 15,000 minutes up to 204,000. In addition, our philanthropy teams brought in enough resources across our network to bring us $1.5 million in iPads, which were delivered all across the enterprise so that our patients could get the care they needed without having to leave their homes. We also are very active in several research investigations. So 7 of our acute care hospitals are participating in the remdesivir trial now. Of course, as of May 29th, many of us are going to be cut off of those trials, except when we want to enroll our obstetrical patients. But we have active patients, and we've actually cared for COVID patients here in 4 of our facilities and maintained their lives through the use of this. We are regularly monitoring the outcomes. Since we have many convalescent patients here, we're collecting their plasma. These, as you probably are aware, the plasma is rich in protective antibodies, and so we can treat up to 4 patients with the plasma from 1 patient. So our research division is monitoring the outcomes of this treatment. In addition, our infectious disease clinicians are partnering with biotech companies here in the Bay Area, as well as others across the country, to really understand what the ongoing clinical impacts are. And Dr. Malhotra has already explained many of these trials that are going on, and we are actively engaged in that. Lastly, we have several medical epidemiologists here at Sutter Health in our research division, and they're partnering with the Institute for Health Metrics and Evaluation at the University of Washington to try and understand what the modeling will be like. What can we expect to see in the future? Will we have, the peaks and valleys experience as we get from fall into winter? Will we have the fall peak and then it trails off? Will we experience the slow burn? There's a lot of different models out there. Obviously, there's no model that's perfect. Some of them can be helpful, but it's really important that we look at these different models to anticipate what we're going to do down the road. In addition, as Dr. Labraccio said, we have a very active equity division within our research department, and we are looking at specific neighborhoods within zip codes to see if the residents of those specific neighborhoods are actually faring better or worse. You can imagine the Berkeley Hills versus East Oakland may show Alameda County when it's all summed up, but there may be different outcomes as we look at those different neighborhoods. And then lastly, what about coming back? we're using, like I'm sure many other healthcare systems are, a very phased approach to broadening services. It's based first on safety, because if we can't keep our employees and clinicians safe, we can't keep our patients safe. We will be gradually broadening our services so that we can follow all of our local guidelines. Public health officers have specific orders. We operate in 22 counties across Northern California. And so it does give us a little bit of challenge, because the health officer within each county can have somewhat different rules than the state does. We also use universal masking for all patients, temperature sensitivity. You've all heard about these strategies that we and other systems like Dr. Labraccio described very well at the start to monitor our patients and our staff. I think it's important that all healthcare systems remind patients who are out there, those of you in the audience that are patients yourselves, that we want to help you get through this. We are safe environments. It's important you understand that we have done incredible things to enhance the cleanliness. We increase the frequency. We use special reagents now. And it is really important that you see us as a safe place to come, because as we learned, the number of background comorbid conditions you have increases your risk for a poor outcome. COVID will be coming back to us in the fall, and we want to be ready for that. So if you're a diabetic patient, now's the time to get in and be sure that your hemoglobin A1C is in good control. We know from data that patients whose A1C levels are more out of whack than others do not fare as well. So it's important that we do the health maintenance functions now. Your clinics are safe places to come, and it's important you see us. And then lastly, throughout all of this, our emergency services have remained open and accessible. And please don't ever think it's a wise idea, if you're having chest pain or symptoms of a stroke, to think that a hospital is a dangerous place. We're probably one of the safest places you can come, and delaying that care could have severe ramifications. That's the end of what I'd like to offer for today, and I'm very grateful to have been invited to participate. Thank you.
Thank you very much. So let me continue to share from this end here and introduce the next panelist. We are very glad to have here today with us Dr. Atul Malhotra. She's the Dean of the School of Medicine of UC Davis. Dr. Atul Malhotra was appointed as Dean of UC Davis Medical School in April 2019. She oversees one of the nation's top research, academic, and medical training institutions, ranked the #7 in the nation in primary care. As an international recognized scholar in rare neurological disorders, Dr. Labraccio leads a school that's at the forefront of COVID-19 testing and the research advancements. Dr. Labraccio, thank you very much for coming, and let me stop sharing here so that you can share the slides from there.
Thank you very much. So I'm very appreciative. Thank you, Dr. Leal. First of all, I just want to echo what Dr. Eisenberg said. You know, I'm a neurologist, and so we want to make absolutely sure that patients are coming to the hospital for all the acute things that they need. You know, our cancer doctors, and I've been here 24/7, and I can't say enough about people needing to take care of both their chronic and acute illnesses. But I want to tell you a story about what the School of Medicine did and how we partnered together with our researchers on the UC Davis campus and the School of Medicine, the School of Veterinary Medicine, and the hospital. And one of the things that really made this an opportunity for us to work together. And so as Dr. Labraccio mentioned, on February 26th, we had the first community acquired case. And that really gave us pause about how we could really leverage the talents of the researchers, the clinicians across the entire UC Davis enterprise. So on March 2nd, we had a meeting and we brought everybody together, including the doctors taking care of that patient, the pathologists who were working to develop a test, the basic science researchers, virologists, and we put our heads together. And out of that came several initiatives that I'm going to talk with you about, but it truly has been a collaborative adventure and really brought everybody together and allowed us to give cutting edge treatments. We also initiated a 7 a.m. call where we had 100 different clinicians and researchers and administrators all together on the phone. And when we did that, we were able to do several things. We were able to bring to our patients within about 5 days cutting edge treatments. Those were clinical trials, several of which have already been discussed. We also were able to take the sample from the patients we cared for and work together within days to get that virus sequenced, to take it over to our main campus and study it, and then use that virus to develop our test here that we were able then to launch about 14 days later. That really led to a collaboration, not only for the basic science, but also launching several ongoing initiatives, including work in the California Primate Center and additional other trials. We were able to actually say to our investigators, we will help support you to do research in COVID beginning on March 2nd. And we supported, together with the UC Davis Provost and Office of Research, several $100,000, and we funded 220 grants to then allow researchers to get the preliminary data, and have already submitted funding to several NIH agencies. I was so pleased to see Francis Collins in your introduction. He's been a tremendous supporter of moving actively for COVID research. Currently at UC Davis, we have 24 active COVID studies for patients, and we've created a COVID research fund. But a picture's worth 1,000 words. This is a picture of the UC Davis Medical Center when we were, I think, on the front page of most every journal and TV channel on February 26th, 27th. My office in the education building looks out over the parking lot, and there were 5 or 6 trucks every day there for weeks. The team actually took this and ran with it. So we published our first community-acquired case. That patient was treated in a compassionate use protocol with Stu Cohen, leading to a paper in the New England Journal, and subsequently for the work that Dr. Fauci's been talking about with remdesivir, UC Davis contributed 19 of the 1,000 patients to that trial. But we were able to leverage that work into developing our own test. So that was the work where we worked and partnered with the virologist, and we were able to develop our own test. And now we've expanded that testing. This is one of the machines that you see. But really, this is what School of Medicine do. School of Medicines actually take patients, and we do excellent patient care, and we partner with research so we can advance care. And that, I think, is really one of the things that schools of medicine across the country will contribute to the COVID research, really advancing science. And of course, in the middle of all this, we had our medical students, and our medical students were not in the clinical area. We had to take them back and teach them about COVID and how to care for them. They have been doing everything remote, and we're now bringing them back. So we're training the next generation of physicians on what it's like to work in a pandemic. How do you take care of your patients, and how do you take care of yourself and your staff and keep everybody safe? Those students are now graduating, and we are pleased that we'll be doing a virtual graduation on May 29th. And then they will enter their next phase of training in this very unprecedented time. But we believe at UC Davis that they are prepared. So some of the recent works I wanted your audience to know about are 2. One is the convalescent plasma transfusions, and this is part of 39 different institutions who are using this under expanded compassionate use. This is incredibly important because of the hundreds and thousands of people across the country who have been affected by COVID-19. This is the way every single survivor can give back. they can give blood locally. That blood then is processed and can be used to treat sick COVID-19 patients. We have treated 2 patients with this, and they've done well. This is something that's been around for 100 years. It's been done in Ebola and MERS, and we know that in patients this can work. And this is one of the many multi-pronged approach that we're using to treat patients. So it is a chance for people to give back. If they're local, they can go to one of the Vitalant blood donation centers in Sacramento, but across the country, you can simply go to a blood donation, and you need to be symptom-free from COVID-19, and you must have had a positive test and be symptom-free for 28 days. And of course, you have to have all the other things to donate blood. The second thing is something that I'm very proud about UC Davis. At UC Davis, we have a partnership with lots of entrepreneurs, and our faculty are very interested in innovating. In fact, we're going to be, I've been talking about this, we have Aggie Square, which we're building over the next couple of years, but really in the COVID world, we've been having a virtual Aggie Square where we really are trying to bring cutting edge innovation to our patients and partnering. So the company, Verinaday, is in the Bay Area, has developed a unique delivery mechanism, and they are a vaccine company. So on May 1st, they began preclinical testing with the UC Davis Mouse Biology Program. And so that is something that we are working actively here at UC Davis to not only treat patients, but to prevent disease. So this has been a real journey for us since the one patient that came on February 26th. It's truly been a partnership, a chance to showcase not only the incredible clinical care that we've been providing, but to bring cutting edge research to those patients, as well as to really think proactively on how we're gonna fight this disease in the future and bring vaccines and other things to the forefront. And I think that we're uniquely positioned to do that with our resources and our world-class veterinary school of medicine. So it's really been a wonderful partnership. I'm so pleased that the Sacramento community is doing so well. And I can't say enough to echo what Dr. Eisenberg said about the hospitals are the safest place to go. This is a great time to get care. And we absolutely want people to get the care they need. That is incredibly important. As a clinician, a neurologist, I can't say enough about that. So with that, I'll conclude. And I thank you very much, Dr. Leal. It's been a real pleasure, and I look forward to hearing the rest of the presentations.
Wonderful. Thank you very much. We will continue now. We have another medical doctor as a guest here, and I'll let him introduce himself.
Sure, I'm Dr. Atul Malhotra. I'm a pulmonary and critical care specialist. I practice in Southern California in the Inland Empire in the Corona and Riverside area.
So in early April, you had 35 years old patients, is that correct?
Yes. Yes.
Let me ask you, how's the patient doing?
The patient is actually now doing well. He was very sick, however, when he first came in. This was a 35-year-old patient without any medical problems, came in with just a 3 to 4 day, actually not even 3 to 4 day, just a couple of day history of cough and some fever and flu-like symptoms. When he came into the emergency room, he had some infiltrates on the x-ray, but was really not very sick. He was requiring only 2 liters of nasal cannula oxygen. I saw him in the emergency room, admitted him to the hospital, but in less than 12 hours, his condition deteriorated. His respiratory status got worse. We had to transfer him to the ICU. We had to intubate him and put him on the respirator. And since then, he was, and then his condition continued to deteriorate. He required high FiO2 and high PEEP. Now that we did several maneuvers, did start him on some medications. Excuse me. We also did prone him. We were able to transfer him to UCLA from Parkview Hospital so that he could get some experimental medications. He did not get the remdesivir, but he did get the interleukin-6 inhibitor. He subsequently required a tracheostomy and subsequently was able to be weaned off of the respirator, and actually just today his tracheostomy was decumulated as well, and he is doing quite well.
So he was a 35 years old patient that came from Mexico. So that's where he acquired it.
Yeah, he actually went to Mexico. He lives here in the United States, but he had gone to Mexico for 1 day.
All right, so he'll come back later, and you don't want to miss the second part of his interview that he's going to tell another story. Another guest that is going to be here today asking questions, and I know that you have many questions in the chat now, questions about the antibody test and things like that. We are going to address all these questions, just be patient. So let me introduce the person that's going to be asking also questions. He's a former NASA astronaut.
Hello, I'm Steve Robinson, professor of mechanical and aerospace engineering here at UC Davis and former astronaut. And I have a few questions about the coronavirus and COVID-19.
All right, so Steve is going to be asking questions, and before we get on the debate, on the answered questions, let me give you a very short, brief review here, what we have developed between the Symposium 1 that we had on April 23rd and now the May 14th. So lots of developments. Let me go back again and show this structure of the virus here that you know very well. And the most important part is the genetic material, the RNA, that's hidden inside of this envelope. It's surrounded by this envelope is this yellow part here that I depicted, that you need to wash your hands with hand sanitizer or with soap, like Dr. Mavericks just mentioned, and try to get this envelope destroyed, because this virus has no value if this envelope is destroyed. The envelope is that protects the virus, and the virus gets to your body. There is a receptor that is in the human body that takes care of this virus entering the cells. The virus has to hijack your cells to be able to replicate. So there is a receptor in our body, and we're going to hear a lot about the ACE2 throughout this presentation today. There is also another one that's called temperature. You're going to be listening about temperature. Everyone seems to agree that the main route of getting into the cell is through ACE2 receptor, but there is also the role of temperature that's going to be mentioned a couple of times here. What's ACE? ACE is the receptor for the virus, but actually for us, it's an enzyme. It's a membrane-bound enzyme that plays a very important role in our homeostasis. It's an enzyme that degrades, that changes angiotensin 1 into angiotensin 2, and this is very important for homeostasis, like I said, for control of blood pressure and the other very important function in our body. So these things are moving so quickly, and with the advent of cryo-EM technology, the structures of both the spike protein that sticks out of the virus, as well as the receptor, have been resolved. This is the structure of the protein, the spike. The spike actually has two parts, two domains, the one that's in pink and the other domain, and the one that is in golden is the receptor bind domain. When they bind, they separate the hot pink, that's this S2 domain, and that's what facilitates the entrance into the cell. This is the receptor. It's amazing. 1 month, and we have so much publication, Nature Science, and the cell, and show the structure of these receptors. This one here, the receptor is determined the structure together with the amino acid transporter. And here, the structure of the receptor bound to spike, only the receptor domain shown here in the golden part, and the rest of spike is removed so to make the picture a little bit more clear. So spike has these three parts of the binding domain. One of them sticks out, binds to the receptor, and avoid any contact with the rest of the protein. So protect the rest of the protein. Another development that was very important for us, nowadays people, before they publish the paper, they submit for these preprint servers like bioRxiv and medRxiv. And this is one of the highlights that I have seen recently is this paper by single-cell RNA, where they show that 83% of ACE expressing cells are alveolar epithelial type 2 cells. This is very important because it's the alveoli where everything is taking place, the change of oxygen. We have more than 500 million alveoli in the lung, and what they are doing there is just the exchange of carbon dioxide and the oxygen. So you are taking oxygen and deliver that oxygen to your blood that's circulating outside of the alveoli, and that exchange of carbon dioxide is taking place by hemoglobin that are in the red blood of your cell. What happens when you get the virus, you get these inflammations and destroying the tissues, and there's separation, and there's a big layer now of liquid pools and other things that are preventing or making more difficult this exchange. So the blood is still circulating, the heart is doing the work, but the oxygen is not getting to the blood vessels, it's not circulating in your blood. So what happens with that is that we get a lower reading of SpO2. And with that, you have some problem that comes for the oxygen saturation is not there. That I'm going to move to the first question that we have from the chat here. And that question goes to Dr. Atul Malhotra. If I am caring for a possible COVID-19 positive person at home, and I observe that the oxygen level is decreasing, at what saturation level should I seek medical assistance and or take the patient to the ER? Also, I would add that a more recent question here. This question asks, could you comment about silent hypoxemia? Dr. Malhotra.
Yeah, so it's a good question in terms of what oxygen level should be a source of concern. We typically look at a saturation above 90, which corresponds to the PO2, a partial pressure of oxygen of about 60 millimeters of mercury, as being normal. So if it goes below that, it's abnormal. But what I usually look for in this context is a trend. If you see a oxygen level that's deteriorating, if it goes from 97 down to 93 or 92, that to me would be cause for concern. So any saturation below 90 for sure, or rapidly deteriorating oxygen level would be a potential trigger. But it's more than just the oxygen. So if the patient overall is very sick or doing poorly, I would sort of take those factors into account as well. And typically being checked out in the emergency room is a good idea. The question about happy hypoxia is a very interesting one. We actually put together a panel of 10 or 15 of the top people in the world interested in control of breathing. And we all had slightly different opinions about what exactly is happy hypoxia. So we're currently working on a document to describe what is happy hypoxia. And so the short answer is right now, I don't know. What's being described, though, is that there are patients who have low oxygen levels, and it's a surprise to many people. The patient's not aware of how sick they are, and the doctors are surprised at how low the levels are, even though we're confirming them with arterial blood gases. So we know it's not an artifact or a measurement error. And what we think this is that oxygen itself is not a potent trigger for shortness of breath if people are able to breathe a little bit more. So if somebody has a low oxygen level and they're able to make up for it by a little bit of extra breathing, they may not be aware of it. The doctor may not be aware of it. It can be sometimes a surprise to many people. So this issue of happy hypoxia is something that's probably been around before. It used to be called silent hypoxia, but it's sort of been renamed in the COVID era. Suffice to say, oxygen is obviously important, and it's one of the things we key on when we're managing these patients during critical illness.
Thank you very much. We'll go back to you later with more questions. Let me move to the next question here. What do the deaths in early February imply about where we really are on the infection curve, and how many really have been infected and the real death rates? This question comes from William Jackson, that might as well be our former chair of the Department of Chemistry. What's the status of research on children's role in spreading the virus? And I'll add one more question to this. That is the recent information about the Kawasaki syndrome. And I'll ask Dr. Labraccio to address these questions. And also an additional very recent question is that where children may not be manifesting the disease, but they may be being a carrier and affect the parents and the elder people live with them. Dr. Labraccio?
Sure. Again, great symposium. I really appreciate all my colleagues' introductory remarks. Very informative. I don't think that the deaths in early February are particularly pertinent. We're on the curve that we're on. They finally got it right on CNN after approximately 6 months. The one thing that is not going to change and that does not lag is actually new hospital admissions. And the reason I say that is the following. The number of cases is almost irrelevant because as we continue to test more and more, we will pick up more and more cases. So it's not really, the number of cases is not really great. And we know we missed a ton of cases because we sent everyone home who had mild symptoms. The deaths in early February just simply tell us that, in point of fact, the disease was spreading around our community a little earlier. But when we've tested large populations for past disease, you're using antibodies, the rates of exposure in various communities have been between 1% and an absolute maximum of 5%. That's probably wrong. The range is probably 1% to 3%, except in New York City. So I don't think the fact that we had early deaths mean anything other than that we were not very vigilant, given that this was already spreading around the world elsewhere. We could have been more vigilant. And in terms of the real death rates, even if the current death rate is still approximately 6% of diagnosed cases, the real question that we have is what's the incidence of people requiring hospitalization and the death rates after hospitalization. Those still remain stubbornly high. And no matter how many people we think were infected, the rates compared to the regular flu they're not 60 times higher. The flu is usually 0.1 to 0.2%. So they're not 60 times higher, but they're not the same. So my guess is we're going to settle out in the 1 to 1.5 to 2% range once we really get an idea of how many people truly were infected in each of the places. But it's still way too fatal and seriously too many people who die who require hospitalization compared to other diseases. So I'll stop there on that particular question. The status of research on children's role in spreading the virus, that is a great question for which I do not have a great answer. We really don't know. Here's what we do know, is that there's a wide variability in the level of virus titers in various children. We do know that they have been associated with spreading the disease to adults. We do know that a good deal of the transmission, as has been studied in both Italy and China, occurred through close household contacts, which may have included children. So what we can conclude is what Dr. Fauci concluded. We don't really know a lot, but we also cannot afford to discount their role, and that it is cavalier to just simply open up the childcare and elementary schools without really studying this and really understanding this. We should not assume that just because they may be relatively asymptomatic and less affected, that in point of fact, they are less contagious. So a lot to still learn. But again, they definitely can spread the disease, and we still have to worry about that. In terms of Kawasaki syndrome, it may not be exactly Kawasaki syndrome. It looks like Kawasaki syndrome. What is common is that the disease series that have been reported clearly show a myocarditis driving multi-organ system failure. That is for those who are lay people on the symposium, basically a lack of function of the heart muscle, leading to essentially a sort of very unusual sort of heart failure in these very young children. And it does not appear to be associated with an acute infection for COVID-19. It does seem to occur sometime after, as many of the children do have antibodies to the coronavirus when they've been tested. A lot yet to be learned, but again, this is why we can't afford to be cavalier. We don't know all there is to know yet. And there is no more precious resource than our children. And we definitely, just because they're underfoot and we may be tired of hearing them scream in the background of Zoom calls, is no reason to send them into the storm. So that's my answer. Thank you.
Thank you very much. And now we move on. We have more questions. And the former astronaut, Stevie Robinson, has a question.
Hello. One of the questions is when we wear cloth masks that are often homemade by somebody, we're told that we're protecting others from our own potential infection. But is it also true that we're protecting ourselves from other transmission of virus from other people? If that's true, shouldn't we be wearing eye protection too? Of course, when you're breathing, you're ingesting air, but your eyes are exposed. So how important is it to wear eye protection when you're in public?
All right, so this question of eye protection is very important, and it has been come to us from different sources. For example, Martin Price also asks about he should be wearing glasses or not. And also the issue of conjunctivitis, because the eyes also express ACE2. We had many cases of conjunctivitis in China, but apparently there are not so many cases in the United States. So I would like to ask Dr. Bill Eisenberg to address this question, particularly with the issue of the expression of the disease in the eyes. Dr. Eisenberg?
Yes, thank you so much, Dr. Lau, and thanks so much, sir, about the question itself. I think, you look at the six of us on the screen here, and many of us have glasses on already. We're regular protectors of droplets. And as Dr. Labraccio stated, we think most of the protection has to do with avoiding droplet contamination. As Dr. Leal said, we definitely know that there are ACE2 receptors in the conjunctiva. And in fact, there are lots of good studies that even at a time when the samples from nares no longer contain virus, they can be harvested from tears. So it definitely is a source. It's unclear why it is that the China experience with rampant conjunctivitis has not been seen here in the United States as much. I will tell you, I always wear eye protection when I go out, not just because I need it for seeing as well, but I think it's always a good idea to protect your eyes. Barrier methods are very effective, and maybe I come at it from the perspective of an OBGYN, but barrier methods are used to protect both parties involved. And so I think it's a really good idea that you continue to wear both masks and eye protection when outside.
Okay, thank you very much. We continue then. There's more questions, and questions about cats, whether they transmit SARS-CoV-2 to humans. There are many questions in the same line. I combine all these questions and ask a specialist, for Dr. James Sykes from the School of Veterinary Medicine, and he has a response.
Yeah, so there's still absolutely no evidence that dogs and cats can transmit infection back to people. We know that infections of dogs and cats can clearly occur, and that they can shed virus. And at least experimentally, there's some evidence that cats can transmit infection onto other cats, but those are experimental studies with research animals that might not really reflect what happens in the real world. We know that this virus appears to be best adapted to people and not dogs and cats. And so it may be that although dogs and cats can get infected, they're not able to shed enough virus or shed virus in a way that affects transmission to other animals in the real world. So while it's still possible that in the future we may see transmission from a dog or a cat to a person, overwhelmingly transmission is driven by person-to-person contact, and being close to a person presents a much, much greater risk than being close to an animal.
So cats also, and dogs for that matter, they have SCEs tools, the same as humans and the other mammals. So the virus gets the entry to their cells, but there's more than getting to the cell. There's a whole lot more about that. So there might be that at the end they don't replicate that, you say, in enough numbers so that it doesn't have enough load to go on to transmit. Is that the case?
Yeah, so we know that there's similarities between the angiotensin converting enzyme 2 receptors, that's the receptor that the virus binds to. We know there are similarities between human, dog, cat, ferret receptors. And it seems, based on a number of studies that have been published recently, that maybe the receptors of cats, dogs, and ferrets, it's a little bit difficult to make an assumption about their susceptibility just based on that alone. So if there was one paper that looked at the sequence of the receptor, quite a large portion of the virus binding area, and showed that actually ferrets and dogs were predicted to have low susceptibility and cats intermediate susceptibility. But that is not what we've seen with experimental infections, at least. So it is true that likely other factors are important, and that might be co-receptors, for example, and differences there. And also the host immune response is going to be different for different species as well. The other thing that's being studied is the distribution of those receptors in different tissues. So, for example, one animal host might have more receptors in their lungs, which might make them better at getting respiratory signs and potentially transmitting the infection onto other animals. So those things are we still don't have all a thorough understanding of that, but there's some of the things that are being looked at in terms of understanding the mechanism for different host susceptibility.
Thank you very much. She'll be back with more questions about cats. Steve has a question, and let's see what the next question is going to be.
At times when we use disposable gloves, they're getting hard to get. They are very hard to find. Can they be reused? Should they be washed or cleaned in some way, or should they be left alone for 24 hours? What is your recommendation?
Dr. Monroe Mavericks, could you answer that question?
Sure. So disposable gloves are pretty cheap. If they're in short supply, then we can reuse them. But let me first just back up. Currently, I don't know of any study showing that you're more protected wearing a disposable glove than you're not wearing a disposable glove. The virus is going to have different survival times on different surfaces. On the human hand, I imagine that it could probably last for like a half an hour, but we have lots of antimicrobial built-in defenses that are already in our skin. So I would be very surprised if the virus could survive longer on the human hand than it would on the glove. Again, the virus is not going to infect you through the hand. The virus is going to infect you because you get it on the hand, and then you're going to touch your face, and you're going to self-inoculate yourself. So in that case, wearing a glove is not going to prevent you from self-inoculating. Wearing the glove is not going to give you any sort of protection if you touch a contaminated door handle and then touch your face with that glove. So I would be cautious in having any type of self reassurance that the glove is somehow protecting you. With that said, in certain circumstances, of course, wearing a glove would be highly advisable. For example, if you're going to come in contact with a surface that is likely infected, then you could wear gloves, and then at the end of that time period that you're dealing with those things, you could take it off. You could take the gloves off. If you have problems with hand hygiene, like your hands are just too beat up from all the scrubbing that the CDC is telling you to do, then wearing gloves in that setting would be very, very reasonable. Now, in terms of reusing gloves, absolutely you could reuse gloves. I mean, gloves are inexpensive, so you could easily get 10 pairs. These virus is not going to last forever on the surface. It has a very delicate component. So I would say after about 7 days, the chance of that virus having the ability to infect you would be very low. But to increase the chances of not getting infected, of course, you could sanitize those gloves with alcohol. You could wash them with soap and water. All of these things would decrease the viral load. And we talked about the amount of virus that you're inoculated with is probably directly proportional to the chances of getting infection. So if you could clean up the gloves with soap and water, with alcohol, let them sit for a couple of days, then I think they would be fairly safe to reuse. But don't rely upon wearing gloves as some sort of barrier under normal circumstances. Your hands are probably more resistant to the virus than a surface like a glove is.
Very brief follow-up on that question. did you say that the load that initiated the infection is very important in terms of how the disease manifests? For example,
Well, yeah. So I'm not, I am not sure if there's any studies in humans showing that, because that would be a very difficult study to do. I mean, we can't just infect people with different numbers of viral particles. But from the animal studies, clearly, the more viral particles you infect with, the higher the chance that the animal is going to get sick with the coronavirus, and also the more viral particles you infect the animal with, the higher the chance that the animal is going to get severe disease and die. So absolutely, I think that in humans, it must also be true that the more virus we get inoculated with, the higher the chance we're going to have of getting the disease, and the higher the chance we're going to have of having a more severe disease.
Thank you. Let's go to the next question. The next question coming from Rupali. And they ask, thank you for organizing the symposium. It was extremely helpful last time as well. My question is, how effective are homemade masks in preventing the transmission of COVID-19 virus, especially as the state starts to open partially, how much they can help in preventing the transmission of virus from an asymptomatic person to a health person? Steve Robinson also has a question related to masks.
Fabrics that are being used for masks can have all kinds of characteristics. Are there recommendations on density or porosity or the types of fabric, the types of weave or the material, or are there recommendations of what not to use?
We have a specialist on campus, Dr. Yulo Hisie, and I asked her these questions.
Well, there's no really recommendation for the most effective cloth-made, homemade masks because there's a lot of unknown about how aerosol and droplets pass through, diffuse from the infected person. Secondly, there's a lot unknown about fabric materials. Generally speaking, the N95, the way it works is it has a very thick layers of fibers that are charged. So allow the pores to be large enough, but the charge on those fibers can trap particulates. Common fibers and fabrics do not have that charged nature, so it requires the pore size to control the blockage of any large droplets or aerosol. The temptation is to find something that you can have access to. Generally speaking, if you find a material that's very densely woven or knitted, the fibers and yarns are very well packed together, meaning the pores are smaller, then there's a higher chance to capture the larger droplets through that mechanism. And also, studies have shown if you use multiple layers, that can reduce the amount of transmission, although the reduction is not in terms of times, 1 time, 2 times, or 3 times. So the most important is we need to pick material that we can breathe through. So that sort of is the dilemma. The fabrics easier to breathe through are also the ones that do not block as much of the large aerosol or droplets. Homemade masks do not prevent transmission of COVID-19 viruses. It will reduce the amount of quantity and the size of droplets, but do not block the transmission.
Can you explain what the N95 means?
N95 means the masks can block 95% of particulates that are 0.3 microns or larger, meaning that anything smaller will pass through, and even larger, 5% can still possibly pass through. So the way N95 fibers trap those droplets is by having charge on those materials. So they attract particulates.
So there are additional problems with masks. This is the proper way of wear a mask. And this is not the proper way of wear a mask. So the nose is exposed in here, and it's another route of getting infection. The same thing in here, even worse, when we wear a mask like that. And you have seen this thing so very commonly these days. I wanted to remind you that you don't want to miss the second part of the interview with Dr. Mashehwari. His story is very interesting, and I'm going to bring that later on. But right now, I'm going to move to Stevie for another, Dr. Stevie Robinson, for another question.
Given the conflicting advice that we're getting from many sources in our world today, how does each person decide how safe it is to go back to some portion of their normal life, either on campus or out there in the world? What data should we count on the most? What do you think is most reliable?
Dr. Labraccio, could you answer that question? Dr. Labraccio there? I think we lost him.
No, I'm here. Sorry, it just took me a second to unmute myself. Which tests, huh? That's like an hour symposium unto itself. But it's very clear the following, which is, at least at UC Davis, you can be assured that the test that we're using for viral RNA, the PCR test, has the highest sensitivity and specificity that are available right now across the world. And we purposefully were not the first into the marketplace offering those tests, but we are, I would like to say, the best. And super kudos to Lydia Howell, our chair of pathology, and Dr. Nam Tran, the head of our molecular biology lab. So that is the best test. Unfortunately, it still requires a nasopharyngeal swab, which is, to be quite honest with you, quite uncomfortable. And so the idea of self-administration of a reliable test right now eludes us. I know people are working on a saliva test, but it has not proven to be as accurate. And any time that you have the levels of inaccuracy that are seen, for instance, with the Abbott diagnostic test, you get what happened in the White House. That is, you test people, you miss people, and you can allow COVID-19 to spread among a close circle that are not taking the appropriate precautions because they have false information. And this is really important as we get to the next test, because false information lets you assume risky behaviors that is simply not indicated. So had the White House been practicing appropriate physical distancing and masking and rigorous hand hygiene in addition to daily testing, they would have been better off. Had they been doing daily testing with a more sensitive and more specific test, they would have been better off. I am very confident of the test that we're administering here for our employees and for our patients. And I'm happy to say, at least in Sacramento, we continue to see a very, very low positive rate. And there are no asymptomatic patients that we have tested yet that have been positive for the virus. So those are all good news. The second thing is about antibody testing. And I will just say this, that again, there is a ton of tests out there. The ones that we are running right now, at least the way that we're running them, I have been told are 100% sensitive and 100% specific at 14 days. So the thing is, we can only run a couple hundred a day right this second. We are looking within the next few weeks, because we have a new machine with an even better test that's a little faster, that we should be able to scale up to about 1,000 tests a day. And once we have a test that is 100% sensitive and 100% specific, we then know for sure we're not giving you bad information. Most of the antibody tests out there are, well, first of all, we don't know what having antibodies mean. It doesn't necessarily mean that you're immune. We don't know what level of antibodies confer immunity, even if there is immunity possible. And we don't know for how long that immunity will last. So the idea that you might have a positive antibody test means nothing given the gaps in knowledge we currently have. So we must treat ourselves, even with positive antibodies, as being potentially susceptible to catching the coronavirus until we have more information. I'm very proud to say that Dr. Larissa May, Dr. Stu Cohn, and Dr. Brad Pollack from our emergency room, our head of infection prevention and the head of public health, have joined together to look at a longitudinal study that will really give us those answers. And then we'll be able to say something about what the best tests are and how frequently to do them. And I'm going to close with one other thing about best tests. You hear on CNN all the time, oh, we've got to be doing millions and millions and millions of tests. Again, I believe this is misguided. I don't believe that going out and testing large populations where almost everybody's going to turn out to be negative is affordable, practical, or operationally possible. We need to be focusing on squashing, as we did here in Sacramento, the curve, limiting the number of people who actually have the disease at any one time, aggressively contact tracing people, testing in concentric circles in a serial manner to make sure we are missing no one, and quarantining and isolating anybody who is positive away from their family so that they stop with the constant contact and the transmission in their familial units. This is the way to deal with testing and the coronavirus, not the way that we have been doing it, and not the way that, frankly, you hear from the federal government about, oh, we've done 8 million tests, so we must be great. That's incorrect. We must test correctly and test appropriately. You still need a lot of tests and a lot of availability, but it just shouldn't be a wild gunshot. All right, I'll stop my testing diatribe now. Thank you.
Okay, and we start on testing now. We continue then to dwell on testing. So there are various questions about testing. And these are 2 questions that are representatives of the large number that you received. Regarding false negative results for the COVID test, what percentage of tests are incorrect, and why are they so many false negatives, and what's been doing to prevent that? That's a question coming from Susie. And another question that for reopening communities, what test is more important, antibody or active presence of the virus? So let me very briefly here explain what the difference between these different types of tests is. So we know from the previous studies that when the virus replicates, they start an infection, the amount of RNA from the virus start to increase, the titer, and the amount of proteins associated with the virus also start increasing, and then later on fade away. So these are the so-called antigens, like a spike proteins and other proteins that could be detected. And the antibody is starting being produced later on. We don't know for sure when did that start, but one thing that everyone agrees is that the IgM start earlier and goes and it doesn't go to the very high level, and then IgG comes later and stay longer. And like Dr. Labraccio said, we don't know if we have these antibodies, it is still going to provide immunity, because one thing is having the antibody, another thing is whether they are going to be neutralizing the virus. But one thing that's debatable now is how many days from the time of the start of the infection of the end. But let's say that these 10 days for the sake of explanation. So during this time in here, before these 10 days, we don't want it to be looking for serological tests. We want to be looking for nucleic acid tests. And that questions answer the question from Chuck and Harris, whether this NTA is better. NTA is the nucleic acid test, so that the nucleic acid test is better if you do that at the time of the infection. And then the serological tests come later, but which one is better to do depends on when we are looking at that. If you are looking post-infection, we may want to look at the serological tests. How these tests are done, very briefly, the famous or infamous picture here of a collection from the swabs, and then this material is analyzed. And what I'm looking here is a picture of the genome of the virus. So this is the entire genome, the sequence of RNA of the virus. And as you see here on the right end, there is N, that means that protein that protects the RNA inside of the envelope. So CDC designed primers for that region, and with that primers are a sequence of the genome that you know that are there, and you look at these primers to see if you find those sequences. And if you find that, you get a result that's positive, then we conclude that the virus is there. But look, we are not looking at the virus, we are looking at the RNA of the virus. So it might as well be that you just detect that, but the virus is not viable. There are so many reports of detection of PCR positive from feces, from water, but the question is, are those viruses viable or not? If you get for a person that is sick and you get the positive results, then we can conclude that yes, this virus, this sequence comes from the virus that is very active at the moment. but the other way around is very difficult. Remember the controversy that we have with the virus from the results from South Korea. South Korea detected this quote unquote reinfection. And later on, it was determined that actually this was leftover of PCR RNA that was detected by PCR. But actually, according to Dr. Oh Moon-dong, who is the chair of that commission, these were not viable source of RNA. So of the virus. So these are pieces of RNA. It happens a lot. So the tests are not perfect. It's the best, this is the gold standard during the infections, the qPCR test, but there are also detection of things that are not viable virus. One example that happened here in California, particularly in the Yolo County, is the case of Marilyn Stebbins. She got the disease, and for a long time, and she was here last time, she was getting positive results, even if she had no symptoms anymore. So let's get an update what happens to Marilyn Stebbins. Dr. Marilyn Stebbins is one of these patients that have been having positive and negative results. Marilyn, could you summarize what's going on?
Sure. So my first positive result was when I was hospitalized on March 3rd. Then just before discharge, the day before discharge on March 9th, I was tested again and was positive. On April 16th, I was tested and April 2nd was tested again and was positive a month after my first test. And then on April 16th, I tested negative and was going to do my confirmatory test on April 20th, and it came back positive. Then again, a week later, tested on the 27th, negative. Then tested 2 days later, hoping to get my second negative on the 29th, came back positive. And then again, started the process of 2 tests, trying to get 2 negatives. And on May 4th, tested negative, tested again on May 6th, and voila, I made it, negative again. So finally got my second negative on May 6th. So I've tackled that battle.
And she started another one that she might have a chance to talk about that later on here. So with the serological test, there are 2 types. There's the so-called lateral flow assay and the lab-based test that's normally called the ELISA, enzyme-linked immunosorbent assay. And these lateral flow assays, they are very simple, as like a pregnancy test, but as you see, they fail in sensitivity and specificity. So these are tests that are in the market now. These are the 2 top tests that based on the FDA homepage. So what this test do is that, for example, here, this is an example of a test from a friend from Sweden. They just sent me this result here. This put his blood there, put a little solution, and let eluted, and later on look for see if there was IgG and IgM in his blood. And he gave this negative results because there's no bond there. Now the question is that, is this a false negative or real negative? That's the very important questions about the sensitivity of the test. Maybe this test was not sensitive enough to detect even if he had the antibody. Another flip side of the story is the false positive. Dr. Labraccio talked about that. False positive indicated that it's not specific to the antibody for SARS-CoV-2. For example, a person may detect, like I put in here, 2 bands, and think that he's a positive, has the antibody, but in reality, this test may be detecting antibody from another virus, not SARS-CoV-2. And the person may get, quote unquote, an immunity passport, but it does not have immunity to SARS-CoV-2 because the result was a false positive. And like Dr. Labraccio mentioned, even if there is a positive, neutralized antibody are working or not. Having said that, and I think that's clear to you now, what's the difference between these two tests, let's move to another question that comes from Jacqueline Ball, and Jacqueline is asking, and I'm going to ask this question to both UC Davis and Sutter. I will start with Dr. Labraccio. Now that the FDA has approved the Roche antibody test, why doesn't UCD Medical Center use it? And the second question is that my doctor agrees that I have the COVID-19 at home, even though I was not tested when I would have tested positive. I am thankful over the worst of it, and now I have a C19 negative test. It concerns me that I am not in the statewide positive count. Is there ever going to be an effort to count folks like me? So let's start with Dr. Labraccio, especially for the first question.
Sure. So, well, we are using it. And as I mentioned, we only have a couple of hundred tests a day currently, which we will ramp up shortly to 1,000 a day, shortly meaning within a couple of weeks, most likely, assuming that the new test that was released by the FDA is validated and is working as it should to be very accurate. I would say the following about, so the research, because we, as Dr. Leal was saying, we really just don't know what it means to have antibodies. Are you really immune? For how long does immunity last? And can you be reinfected despite having some degree of immunity? We know none of these answers. So by simply telling people that they have antibodies, we're not telling them anything that would guarantee that they were no longer susceptible. And so for that, we have to be very careful. Again, because people take that as a license to go out and ignore the social distancing and other rules that they still should, for their own health, observe until we know more. So I would just say this, that we are planning to use it. Our goal is to test all of our employees. We're starting out, and we first started out, just for the record, by testing everybody we knew who had COVID-19 with a positive COVID-19 test, because we expect 100% or nearly 100% of those to have antibodies. That validates what we're doing. And then to move on to the few people who had repetitive exposures, those people in our medical ICU, those people on the one floor where we put all our COVID-19 patients, all the nurses and other workers there, and the people in our emergency room. And once we're through that, then we will open it up more widely. But we're not only testing people, we are doing what we call serial testing. That is, we're looking at the level of antibodies in their blood. We're looking at the time course over which those antibodies go down or up, but most likely down. And then we're trying to also follow all of these individuals for a long period of time into the fall flu season to see if any of them become reinfected, so that we can actually answer the questions that remain out there. And so we're using our testing capacity in a way that we think will really change how people approach the disease. I know that it would reassure people to get tested, and certainly people like you who've been tested would like to know for sure, and you deserve to be tested, and we want to help you get that test at the soonest possible moment.
So let me ask you, Dr. Eisenberg, what's the situation with Sutter? What kind of testings are being allowed and they've been done at Sutter?
Yes, well, thank you very much. And much of what Dr. Labraccio has said is the tactic we're following here. We are, of course, using the antibody test to screen our potential convalescent serum plasma patients. It's really important that we demonstrate that they do indeed have the antibodies. And so that's one use we have. And then we're also testing our positive patients and those that are in units where we know we have many COVID positive patients. I think the only thing I would just like to stress on top of what Dr. Labraccio said is it's so important that people who do get tested do not think that this is a get out of jail free card, that somehow having positivity means the same thing as saying, yes, I can see measles antibodies, or I can see hepatitis B antibodies. Those antibodies, after you've been vaccinated, are protective for you. But we have no idea at this point the value to being antibody screen positive right now.
Thank you very much. Before we move to the next question, looks like Dr. Malhotra has a comment about the false negative. Is that right?
Yeah, I just wanted to add to what's been said, because I agree with everything said, and certainly there's issues with the sensitivity of the test once it's in the Petri dish. But there's also questions about when it's sampled. So we know this is a lower respiratory tract pathogen, And so there's plenty of cases of positive x-rays or CAT scans that are negative in the nasopharynx, and then they turn positive several days later. So depending on timing, you could get a false negative for somebody that has lung infection, but not yet in the nasopharynx. And then the other issue that somebody brought up in the question box, which is also important, is where are your samples? If you do a nasal swab as opposed to a nasopharyngeal swab, you can miss the virus. And so there's false negatives for that reason as well. So plenty of reasons for a negative test. And I'm not that reassured if somebody has a negative test that they're not contagious. It's the kind of thing you have to keep an eye on and still do social distancing and hand hygiene, all the rest of those things.
Thank you very much. Very nice comments. And there's so many unknowns that we have to be very careful. Let's move on to the next question. For reopening communities, what test is more important, antibody or active presence of virus? I think the person, Lynn, is asking whether you should have a serological test or qPCR test. Dr. Mavericks, would like to comment on this one? May I add a second one that just came in as well this morning? This person had everything that seems to be a COVID-19. It was not tested because at that time it was not available. And then after some time, that person was tested by serological test and came out negative. And that person indicates that they have a non-Hodgkin lymphoma that's affected the B cells. And whether that negative could be that because of that condition, the B cells are not producing the antibodies. Is that something that we could address as well?
Sure. So the first part of the question is what type of testing is important? Before I answer that, I want to just bring in one additional point, and that's the pretest probability. So even if a test is highly specific, if the pretest probability is very low, then you're going to get a lot of false negatives. So what that means is that if the test works very, very well, and you go into an area where there's absolutely nobody infected, you're still going to get a lot of positives depending on how many people you test. So there should be some type of pretest probability before you do the test. But in order for us to know those types of things, we should really collect some information on the people we're testing, and that should also be transmitted to the CDC and these California organizations. Right now, what we do is we test and we report the positives, but we don't know anything about the negatives. And of course, at the center, we would know, like in UC Davis, we know how many people we test, but what's transmitted are the positive results. So it would be nice to know that, 100% of the people who tested positive had a temperature above 99. Then we would know that, if your temperature is below this amount, then the likelihood of you testing positive would be very low. So there's lots of things that are not being done that could be done that could help guide us in the future. Of course, if we're trying to reopen communities and we had some type of testing strategy, you'd be looking for active disease. So that would be the PCR test. You wouldn't be testing for antibodies because there's a long lay time before the antibodies come up. And as you said, who knows how long that is, but probably around 10 days. And then with the question about the lymphoma patient, it depends a lot upon the specifics of that lymphoma patient. So if the patient is on treatment, that could lower their ability to mount an antibody response. People who are on immunosuppression will have a lower ability to mount an active antibody response. If the cancer is very widespread, that might also lower their ability to, and this is a B cell cancer, so they didn't tell me what type, they just said non-Hodgkin lymphoma. But it depends on the type of cell. So presumably this person, I forget the name, I'm sorry, who submitted that question has some functioning B cells and probably some naive B cells. But if they're on therapy, it could reduce their chances of mounting an appropriate antibody response. So my response to their question is absolutely, your disease could have been related to your inability to mount an antibody response. The flip side is, were you false positive? And I think probably it's more related to the disease, but we would never know.
All right, thank you very much. So let's continue here. Late in the symposium, we have a patient telling their near-death ordeal with COVID-19, and we move to the next question, comes from Connie and another one from Glenn. I think I may have had COVID-19 in early April, but symptoms were not life-threatening, so I did not qualify for test at that time. Should I be tested to see if I had it or not? And the other question is that, can you discuss what procedures and safeguards are in place for patients who need lab work and the test at remote clinics and need the procedures at the specialty clinics at the remote location? So this is a question that applies to both the UCD Medical Center and to Sutter. I would start with either Dr. Labraccio still there. If not, maybe Dr. Alison Brashear, one of the two, could answer this question for us.
Right, I'm still here. Allison, you want to take this one? I think you're still on mute.
Happy to help. So first of all, the procedures that are in place in our hospital include lots of work on the cleanliness of the hospital. Wear masks. All the people are wearing masks in our hospital. And we actually have very few, as of this morning, 2 patients in the entire hospital with COVID-19, and they are all in one area. Likewise, all of the employees and patients are screened going into the clinics. As I said earlier, I think it's one of the safest places to be, is here in our area in the hospital and our clinics. The other question was about testing. If you had symptoms in the way back, and should you be tested? Again, that goes back to this issue about antibodies. And I think the issue is we can test for antibodies and we know that we can detect them, but what do they need? How high do they need to be? And how long do they last? And we don't know that. One of the things I'd like to make a pitch for is the research protocols that are out there that Dr. May at our institution is leading, is about exploring that and trying to determine about the antibodies. That is what's going to really give us the answer. And that's going to be the groundbreaking work for how we prepare for any vaccine studies. So we're going to need to know what antibodies, when, and how high for a vaccine. So one could imagine a vaccine trial done in animals, and then they will need to measure that. So I think that would be really important. So great question. Thank you for all of your interest. It's really very helpful.
Wonderful. What about Dr. Eisenberg? Maybe the question, the procedures and safeguards right on your alley.
Yeah. Well, I think all of us at different healthcare systems are using the same strategies that Dr. Brashear just described. It's really important that we mask all of our employees the moment they come in, right after their temperatures are checked and symptoms are checked. They get their mask, so they're all covered. They're all wearing protective equipment. We have enhanced cleaning processes, as was mentioned. So you should really see this as the right place to come for these kinds of tests. And I don't think I have anything else to add on the comments about testing, because until we know more about the value of these antibody tests, it's really very difficult for us to give you any confidence that whatever that test result means, you can take to the bank.
Thank you very much. I move on to the next set of questions. That's pertain to something that's related to a recent publication from our colleague Jonathan Eisen and collaborators from the University of Oregon. They look at the airflow in buildings. That's a question related to that Steve is asking, and I think that's a question that is good for Dr. Atul Malhotra. Let's listen to the question first.
Also, for cloth masks, are the masks mostly absorbing droplets, or are they actually filtering air to keep the virus from reaching your lungs and your soft tissues?
Dr. Malhotra, could you answer that question?
Yeah, so the questions about these cloth masks, and to be honest, they haven't been that well studied. There was a paper in the British Medical Journal Open that was published from Vietnam, where they looked at these cloth masks to see how well they protected healthcare workers. And there's two issues. One is you're protecting yourself if you're wearing the mask, or you're protecting others. And this was more looking, are you protecting the healthcare workers if they're wearing a cloth mask? And when they studied it, the relative risk of the healthcare provider getting an infection wearing a cloth mask was about 13. That means a 13-fold increase of getting an infection or getting the symptoms wearing the cloth mask. So they're really not terribly effective at protecting the provider. If you're talking about protecting others and you don't have symptoms and you're just worried about coughing on other people, then they probably are a reasonable way to get the gross particulates away from coughing them on other people. So short answer to the question is the cloth masks really aren't very good at filtering out virus getting into the person wearing the mask. As we heard about earlier, the N95s and other things are recommended for aerosolizing procedures. A regular surgical mask is reasonable for people not in the context of an aerosolizing procedure. But the cloth masks can give a false sense of security to providers, at least in some cases.
All right, thank you very much. So let's move to the next topic of therapeutics. And let's talk about the remdesivir that was mentioned here today by Dr. Atul Malhotra. I'm going to borrow this slide from the presentation of Dr. Antonio Fauci gave at the National Academy of Science last month. And he shows here the big trial that's going on, not only in the United States, but overseas. And the UC Davis is there as part of this trial. And they have including 1,063 enrollees for this. On 29 last month, this Chinese group published this paper of their own clinical trial. Unfortunately, that clinical trial had only 236 participants, and it was not the default. They planned to have more participants, and they planned to have a very high statistical power of 8%, and they were able only to have 58%, which is not very good to conclude a clinical trial. And the reason that they didn't have enough patients because the disease was under control by the time that the study was going on. Yet they conclude that the time to clinical improvement in the remdesivir group was not significantly different to that of the control. Again, it's a caveat that there is no statistical power to make a very strong conclusion here. Another one interesting is that patients receiving remdesivir or placebo within 10 days of symptoms, those in the treatment had a numerically fast time to clinical improvement than those receiving the placebo. Again, I repeated that we can't conclude if there was no statistical power for that. However, it seems to indicate there is something to do with the time of the intervention when you leave this virus. And here's another interesting that we didn't coordinate that, but Dr. Malhotra mentioned about where you collected those. And here's what one of the graphs from that paper. And we see when they collected from the upper respiratory and the lower respiratory tract, these are scales in the log scale. So the numbers are humongously high in the lower respiratory tract. One thing that's kind of raised a question here is that we see that the level of RNA, the virus, is in the control as well as in the treatment. They are not so different. So raising two questions, whether one possibility is that this qPCR might not be the best technique to test that. Another possibility is that if it's working, probably the mechanism could be different of the virus replication, because replication does not seem to be affected in here based on this figure. So in April 29, the same day that this paper came out, NIH gave a preliminary results of this large trial, and that one concluded that they have modest results with the 31% first time to recover than those receiving the placebo. So that was 11 days for the patients with remdesivir and 15 days in average for the patient with the placebo. So let's shed some lights in here. Looks like there is some effect there, and probably because the effect is not so strong with the results from China with a study that was underpowered, probably they could not see that. Now NIH is going to move to the next step, and they're going to use another trial that's going to have remdesivir is going to be the baseline. Basically, it's going to be the placebo is going to be remdesivir, and the treatment is going to be remdesivir plus Olumiant. So that's a combination of the two, and they start soon. So I want to move to this topic of therapeutics, and I will ask both Dr. Atul Malhotra and Dr. Bill Eisenberg what kind of tests are they conducting, what kind of trials, and one of the questions, how a patient qualify for those trials. And one very interesting question is that, what if I get only the placebo? So if you go to a hospital and then we get only the placebo, how do you feel about that? Because you need the placebo there, but for the person that's getting the treatment, they don't know whether they are getting the treatment or the placebo itself. So let's start. Can I start with Dr. Eisenberg first this time? In fairness for Sutter.
Thank you very much. So first of all, we, as I mentioned earlier on, we were participating in the trials for remdesivir at 7 of our hospitals. As I think probably everybody has read, initially those trials were randomized, and so there was the placebo possibility. More recently, the government announced that Gilead said that all of the patients would be getting active drug and no longer getting placebo. But then also another piece was that as of May 29th, there would no longer be the trials any longer. So the drug is now exclusively being managed and distributed across the country based on hospitalization in various counties. So I don't have much more to add other than that right now. It's a little bit difficult to know about how much we're going to be getting here in California. We have had a far better outcome for our state relative to other states. So there may be what seems to us like an unfair distribution of drug around the country. And as far as if you get placebo, that's sort of one of the downsides to being a participant in a randomized control trial. There's always that risk that you will get placebo.
Dr. Labraccio, you
Yes. Yeah, so I think one thing is to step back and think about how the FDA works. So when there's no treatment, it's placebo versus the drug that you want to test. When there is a gold standard treatment, such that's been approved, then it typically is the treatment plus something else. And so in this way, the protocol appears to be a combined treatment. So I think the people listening should really take heart that things move forward in terms of the design of the trials. I think that's really key. We are learning as we go here, and it is great that they were able to enroll so many, over 1,000 patients in the remdesivir trial. The other thing that points out is the large number of patients that are needed to show the difference between two treatments. So that is also key. It's not just looking at 10 patients and saying it works. In order to show how the significance of it, you need large numbers. So my take home would be, as we move and find new gold standards where we had no treatment, it's likely these trials will be against usual care and a new treatment. That's usually the way these work. The second is for everybody to know that things are not answered in small numbers. That's why people need to donate blood for the plasma transfusion trials. That's why it's so important that we get a lot of the answers, because it's not going to be answered in small numbers. And we really appreciate the community, particularly of people who have recovered from COVID, contributing and giving back for the great care that they got. But we're really heartened about the interest in research. I think that is something that's come to light. COVID-19 has really shown the light on the partnership between research and clinical care, and that shows very well in schools of medicine and hospitals and academic health centers like we have in the University of California.
Thank you very much. So the next question, somehow Dr. Malhotra already addressed that question, but maybe has more to add. What kind of secondary infections are associated with COVID-19, and what's the treatments available? And are there other drugs being used under compassionate authorization from FDA?
Yeah, so there's several different questions there. One is about co-infections. And it's interesting, what was reported out of China was about 2 or 3% co-infections. We're seeing closer to 20% in UC San Diego. I believe San Francisco has similar numbers, and I've seen similar published data from other centers. About 20% of patients have co-infections, that is with coronavirus, but in addition, another virus or another pathogen. So influenza is a common one, and we've seen that quite commonly. The good news for influenza is we do have medications like oseltamivir or Tamiflu that is quite effective. The interesting part of that is the earlier you give it, the better. And that's in contrast to what some findings in remdesivir are, where it seems to be benefiting some patients late, which to me is a bit of a surprise. My guess is antiviral therapies will work better early, and some of the anti-inflammatory therapies will work better late. In terms of compassionate authorization, you know, there's lots of things out there. Even hydroxychloroquine, I think, got emergency use authorization, but the data are not very convincing to date, although more than 90 ongoing randomized trials, so perhaps time will tell.
Very good, thank you very much. Let's move to the next question here. I want to remind you that Dr. Maheshwari is going to come later to finish his story. And now I have a question here from Joy Abo, and how can we safely open swimming pools? For many people, swimming is the best form of exercise because it's easy on the joints. Dr. Mavericks, do you have an answer for that question?
Sure. So I agree, swimming is one of the best forms of exercises. I would say that there's no way to know that something is 100% safe. But if you're in a lap pool, then the same types of things would apply to if you're around people. So currently there's no evidence that coronavirus can be transmitted through pools. And if you wanted to go swimming, the first step would be to go to a pool that's very well maintained with chlorine and that appears clean and is not so crowded. These are all things that are important. The second thing would be to see if that pool has any type of requirements on the people they allow in. Like, are they doing any type of pre-health screening? Because even if the virus may not be transmitted through the water, if you're in close contact with people who are not screened before they went into this public place, you might catch it from outside of the water or from the handle or something like this. So there's no way to be 100% protected, but I would say in things that are important activities like exercise and these types of things, if the incidence of coronavirus is low in your area, and if the pool is not so crowded, and if you're going there for exercise and doing a lap pool, so you're not in close contact with other people, you know, as we commonly are when we're in a pool for other type of activities, and you're practicing social distancing, then if you take all those factors in, and if they're doing some type of pre-screening before they let the people come into the pool, it should be pretty safe to go swimming. Of course, you can't. Nothing is going to be 100% safe from here on out, but I would recommend people not be afraid to do things that haven't been really proven to be dangerous yet.
So a question that can follow up on the chat is that whether the filters of the pool, they filter out the coronavirus. I believe it's not.
No, yeah, so it shouldn't be able to filter out. But and I'm not sure. Again, this is probably unstudied area. I'm sorry that I don't know the literature on this, but I'm pretty sure that, you know, we have not dunked people into the water who have coronavirus and then seen if people around them have gotten sick with coronavirus. And I'm not sure if the amount of chlorine will wipe out all coronavirus. But again, the pools are very large. And if they're not so crowded, the amount that you're going to get in you of viable virus should be extremely low. So it should be fairly safe to go in a pool. And like I said, there's no data, as far as I know, that has ever shown community transmitted through open water sources like a swimming pool.
Thank you very much. We sometimes get good questions, and sometimes we get questions that's no good answer for that. That's one question from Stephen Robinson here.
Here's a question with no good answer. What is your recommendation if we see someone on campus who seems to be ill and we're worried about them and their potential influence on the people around them?
So I think that's part of the issue about being a good citizen. If someone is ill, we need to direct them to go see their physician. It is possible, particularly when we get into the fall, it's going to be very hard to determine the flu from COVID. That said, I also would strongly urge people to practice 6 feet apart, physical distancing, and of course wear a mask. But part of what this has brought to light is the role of us all being good citizens in our communities, and that's why social distancing has worked so well. And frankly, that's why Sacramento is doing so well, because we've all been good citizens. So I think that's the big issue here: is practice physical distancing, wear a mask, and if someone is sick and you see they're sick, I think you should speak up, and that person should go see their physician and should be wearing a mask.
We are not following the chat on YouTube, but I just picked it in here, and I see a question about the reinfection, and maybe Dr. Malhotra would comment on that. So this issue, I think, is one of those many questions in this field now that we don't have a definitive answer, but the question is that, is there a reinfection?
Yeah, so the question is, once you've had coronavirus, can you get it again? And the short answer is that nobody knows for sure. It doesn't appear that that's occurring. That case from Korea turned out to be sort of a false positive. There are reports of people shedding virus even up to 6 weeks after an infection, but there's still one primary infection, and then it eventually resolves. There are some data from macaque monkeys that suggest that they do develop immunity, but at the end of the day, people get coronavirus infections all the time from coughs and colds and other things. So my guess is that reinfection is not going to be a major issue, at least based on the available data.
Yeah, I think the question stems from this case that was reported in the LA Times yesterday of a patient that was released and then returned to the hospital, the Desert Valley Hospital. But we cannot make conclusions about the cases, especially N equal 1, right? So like you said, yeah.
Particularly, you can have a false negative than a false positive. You need more than one to draw conclusions, obviously.
Thank you very much. So let's move on. The next question is about the possibility that this virus is also transmitted sexually. And the reason for that question is that because people, as all these curiosities, transmitted so many means, and also because of the last pandemic, we have the Zika virus, and the Zika virus ended up to be transmitted not only by mosquitoes, but it was sexually transmitted. And they end up that a group from Utah, in combination with the group from China, they addressed this question already. So it seems that Dr. James Hoteling already addressed this question in a very recent paper that they are publishing, Fertility and Sterility.
Yeah, so the caveat I would give is that this is, a small cohort of patients out of Wuhan, and we looked at 34 patients and basically showed that in our cohort of patients, we didn't find evidence of COVID in any of these men who had been recovering. They were men who were not that sick. They had generally pretty mild illness, and they were also, generally speaking, about 30 days out from having the illness. We didn't find any evidence of COVID in the semen of these men, nor did we find like a clear molecular way for the virus to get into the testis cells based on single cell RNA sequencing, because the cells don't co-express the 2 receptors that are necessary for the virus to get in. There is another recent study that came out yesterday out of another group out of China that actually looks at a very different group of patients, looks at very, very sick patients. They started with 50 patients, 12 of them they couldn't get samples on because either they couldn't, give a sample, they were intubated in the intensive care unit, or they had died. And of those patients, they actually did find COVID in the semen in 6 of them, but 4 of those patients were actually in the hospital when they found it. So from what we can tell from our paper, our conclusion was always that in patients who are recovering from, mild to moderate forms of the illness who are a little ways out, we don't see evidence of the virus there.
So to recap your paper, you analyzed by qPCR the titers of SARS-CoV-2 in semen, and all results came negative because the CTs were above 40, which is a very high number for CTs. So everything was negative. Then you went back and they asked a question in a different way. You look at the molecular components that are required for the cells to be infected. You look at the ACE2 and also the expression of the temperatures, and you find basically very low levels that would be very hard to explain. So that's the conclusion from the paper, correct?
Yeah, that's the conclusion from our paper. Yeah.
So I see for the publication of the paper that came recently from Yale, where they collect the SARS-CoV-2 from saliva and the higher titer than the swabs. So is it safe to conclude that it's safer to have intercourse than to kiss?
Yeah, I think that would be, the answer is we need more data, right? We really would want to, the ideal study you would do is you would test somebody, you would do a semen analysis right as they were starting to get sick, and then you would just repeat it, every time after, and you would retest. But generally speaking, yes, I would agree with you. We made the same point in our paper and to the media that, if you were having, sex with somebody, the biggest risk of transmission would probably be the respiratory stuff, from saliva or secretions.
Is there anything else that you'd like to add at this point for the public in general, things that they should be aware?
I think the only other component is that, we did find that about 18% of the guys got viral orchitis or some swelling in their testicles. And we don't know, what the exact significance of that is. I think there's 2 separate issues. One issue that's getting worked out by our publication and this other publication is does the virus get into the semen when men are really sick? Do you know what I mean? And then as they're starting to recover, is it cleared? And the answer to that would be, it seems to get in maybe when they're really, from that paper, get in when they're really sick, and then it seems to be cleared, when they're doing better. The other question is, in regards to Zika, what are the long-term implications, right? What happens to sperm parameters, reproductive hormones? And we're actually working with a group, out of China, and I think they're going to collect 300, semen samples, going forward in men who've had COVID. So, I think those are the major points, and more work is certainly needed, and certainly one of the difficulties in our field is nobody can culture human spermatogonial stem cells. So you can't really do any cell culture work, and rodent spermatogenesis is fundamentally very different from human. So it's just difficult to study.
Do you see a decrease in numbers of patients in your regular clinic during this time of COVID-19? And if so, what's the consequence of that?
Yeah, that's a great question. We have basically converted everything to virtual visits. So my team is me, and I have a fellow and then 2 nurse practitioners. And this week we'll see about 130 patients, but those are all virtual. Those are all through the computer. We have had a few patients cancel surgeries we had canceled all our surgeries, and now we're starting to reschedule them. I think there are some patients who are definitely scared, to come near the hospital. Our hospital is probably like your institution, is taking everyone's temperature and, social distancing, and everyone's wearing masks. And actually every patient we operate on gets tested for COVID before we can even operate on them. But yeah, we have seen a decrease in patients for sure. And unfortunately, what we're starting to see is a lot of patients who've put off coming to the hospital when they really should have, and are coming in really, really sick.
Thank you very much for your time.
Thanks. Appreciate it. Take care.
Bye-bye. There is a question that comes here now from Mary Jean, and maybe Dean Labraccio might be able to address this question. How safe is it for schools K-12 to reopen this fall with no vaccine and no herd immunity in place?
Boy, I think that's the toughest question of the day. I'm a parent of a college student and asked that question myself. I think we're going to need to rely on our public health officers to try to make that determination. You know, whether or not the incidence in the community right now, for example, Sacramento has a very low incidence. So the chance of actually contacting COVID with very, very few people who have it. But it is something that I know all of the educators across the country are thinking about. Certainly schools will need to address masking and social distancing. You know, so this is an unprecedented time, and we're in uncharted territory for education. It's going to be a work in progress to get those answers. And I think a true partnership with the medical community and with the educators in the community. So I just want to shout out to all the parents who have taken on the role of educator, particularly those who are going to work, our frontline healthcare workers. That is just an incredible role that you're playing, and we are all so very appreciative. I know that of all the faculty and staff who are working in the hospital, it's been just amazing.
Thank you very much. Let me take one question here of the pet lovers. What if we adopt a cat or a dog from a shelter and the caretaker had COVID-19 and it was asymptomatic, so it was going to work, but it was transmitting, because we know now that they transmit even before the symptoms are there. So I ask Dr. Jane Sykes to this question.
Yeah, so number one, firstly, there still is no evidence of transmission from animals to people. So that's the first thing that has to be remembered, and that dogs and cats represent an extremely low risk to people in terms of infecting them. We also know that some big diagnostic laboratories, IDEXX and Antec, have now tested more than 6,000 samples from dogs, cats, as well as horses that have had samples submitted for other respiratory disease testing, because there's a lot of other different respiratory pathogens of dogs and cats. So these are animals from heavily impacted areas with COVID. They started testing in mid-February, and they've now been testing animals from impacted areas in the US, in Canada, in Europe, as well as in South Korea. And of more than 6,000 samples, all of them have tested negative. So that really, I think, shows you that although we don't know the extent of contact between those animals and people with COVID, infections are very rare, and we would be seeing more of them if spread from humans to animals was a common thing.
So cats, they have their own coronavirus, the feline coronavirus. And what do they do to cats? They get sick?
Yeah, so that the cats have a coronavirus that it's pretty common in young cats, and it either causes no clinical signs or just mild diarrhea. But in some cats, for virus reasons or for cat genetic reasons, that virus actually mutates and enters some of the immune cells in the body and causes a severe systemic disease, which is known as FIP, or feline infectious peritonitis. And that's a fatal disease. Once cats develop it, they die, and they're not actually able to transmit that mutated virus onto other cats. So the cat dies, but doesn't transmit that very virulent virus onto other cats.
Thank you. There are questions coming from the chat now. And maybe I ask Dr. Malhotra if he would be able to answer this question. What's the best way to determine the herd immunity?
Can you hear me?
Yeah, yes.
Sorry, what was the question? Sorry, I lost the audio for a second.
The question was, what's the best way to determine herd immunity?
What's the best way to determine herd immunity?
Yeah.
So herd immunity is referring to this concept that if there's enough people who have been exposed to the virus, that they won't be able to spread it anymore. It's a way of, if you build up enough immunity in the community, that the virus won't be available to spread because enough people will be immune to it that there won't be a vector to send it from person to person if people are immune. That was sort of the experiment that was tried in Sweden, and as best we can tell, so far it's failed. There are many people there who are getting evidence of infections, and there is a problem there. So I think the short answer right now is we don't know. People have written theoretically that once enough people have been infected with coronavirus or with COVID, that the outbreak will be over. But right now, I think that's an assumption. We don't really know that.
Thank you very much. Dr. Mavericks, would be able to answer this question, whether the HVAC system in the buildings are helping spread the virus?
Say it slowly one more time.
So the question is that the HVAC systems in the buildings
like
are helping spread the virus?
Again, I think it is, it's possible for the virus to be in the heating and air conditioning system, but you're going to need to be inoculated with a certain amount to get sick. So the amount that like floats into the system and gets circulated around and somehow comes out of the system, those systems also often have some kind of filtration unit, will probably be very low. And so the ability to get sick from such a low inoculum is probably going to be minuscule. Of course, anything's possible. But I would think that it would be very low.
Thank you. It would be wonderful to have Jonathan Eisen here. He's to work on that, but he's very busy teaching one class. He would not be able to attend anyway. So let me get one more questions of pet owners. The question is that they are avoiding to go to the hospital to take care of the pets. That's a question that Susan is asking. And I believe that's a question related to what we see in the medical field in general, what these things happen in the med hospitals.
Yeah, it's a good question. It's a little bit difficult to assess for us right now because we did reduce our operations significantly to emergencies only. And we took all of our students out of the clinic, so we reduced our services because of that. Other practices, though, are reporting different things. So other veterinary practices, for some practices, business is actually up because people are spending more time with their pets and they're taking them in to their veterinarians, which are considered essential services for preventative treatments and vaccinations, for example. Other clinics in other areas are reporting decreased visits. So, I think it probably is a bit regional.
Is there anything else that you'd like to add for people that are anxious and asking these questions all the time about their pets?
Yeah, I think the main point to make is that, again, we don't have evidence that cats and dogs spread this to humans, that they can rarely be infected. And so what people need to be doing right now is enjoying the human-animal bond. Everybody's kind of stressed right now, and having a companion in the house is very beneficial for mental health and wellness. Actually lowers blood pressure and reduces cardiovascular disease, which are risk factors for COVID-19. So I think they should be focusing on enjoying the relationship with their pet rather than being afraid. I think if they do have COVID or are known to have COVID, then there are precautions that the CDC recommends, including wearing face coverings when interacting with your animals and minimizing interactions with your animals, ideally having someone else in the household take care of that pet just to reduce the chance of transmission, and not doing things like allowing animals to lick you in the face and lick wounds and share food dishes and sleep closely in the bed with you, because especially for immunocompromised people, there are also other infectious diseases that can be transmitted through those mechanisms. But overwhelmingly, they should be enjoying their companionship with their pet.
Thank you very much. Thank you, Dr. Labraccio. There's a follow-up on that question about K-12, and the follow-up is that what about college, with people coming from all over the country and the overseas as well? And it looks like they are giving you all the difficult questions today.
Yes, and I don't have an answer for that one. College is a very crowded place. I have a rising junior in college, and it's something I think about quite often. The colleges are going to need to think about masking, social distancing. I know that the leaders at UC Davis are considering all the different things. Whether or not some colleges will be able to accommodate large classes. You know, I have to applaud the educators across the country who have really jumped into teaching by Zoom. I know my two children, both in college, are doing classes via Zoom. So I think the guidelines for all of us are physical distancing, masking, staying home when you're sick, and isolating. Video visits are excellent because, you may not know what you have, particularly in the fall. So, it could be the flu, it could be something else. The ability to test is key for our community. And that's one of the things I really want to applaud our community. We've been able to get testing. And so testing symptomatic individuals to know whether or not, when they're symptomatic, if it's COVID or something else, will be very important in terms of then doing contact tracing and such. So it's going to be a new world out there for everybody, including all of our students, medical students, college students, K through 12 students, parents, all of us. It's going to be a new world, and we're going to need to adapt, and we're really going to need to learn how to communicate and share information in an active way, just like was done today. I just applaud Dr. Leal about how this has gone, because he's had so much information here, and information is key towards preventing future disease.
Thank you. Dr. Eisenberg, maybe you are able to address this question. I'm going to read it a little bit slowly here. It's unusual for me. My parents are fragile with compromised autoimmune issues. 12 years ago, mother had kidney transplant and is currently taking anti-rejection drugs. Father had stage 3 colon cancer and is cancer-free. With all precautions, gloves and the mask, social distance, and CDC requirements, is it safe for the daughter who lives in the home to enter the general public and return to home? Daughter lives in the same house, hold the practice social distance, has own physical space, room, bathroom, sitting room, etc. Share space in kitchen only. What are the risks and is it safe? That's a very important question.
Well, it's a very important question, and it's a very difficult one to answer. Clearly, both parents have some significant complicating medical conditions that put them at higher risk, immunocompromised status, comorbidities, and yet it sounds like also the daughter is an important person in their lives, somebody who I would imagine is also involved in keeping them safe and healthy. So it's hard to say, well, no, she shouldn't be there, because somebody likely does need to be there. I don't think the parents would be well advised to go out and do their own grocery shopping. They shouldn't be going out of the house and be exposed. So this is somebody who should definitely be practicing all of the habits we've talked about, social distancing, hand hygiene. But it seems like it's important that she also be in the home with her parents. If it were me, I would probably be using my masking within the house with my parents, just because there's no way I could avoid going out into the public. So I would be acting as though I were a healthcare provider when I was around my parents within 6 feet for longer than 10 minutes. I'm interested to hear what my colleague physicians have to say about that, though.
Anyone else want to add something?
Yeah, I agree with what you said. I really think when people are more susceptible, be it immunocompromised or age, we need to be very careful about what we bring to them. This is a time for using the physical distancing and exquisite hand hygiene, et cetera, with that. They are most at risk, and they're the ones who are most likely to get the sickest as well.
Thank you. Thank you very much. Let me present now the part 2 of the story of Dr. Anup Maheshwari, and here it is the story. Now, the other side of the story, you got sick with the coronavirus, probably acquired from that patient. Did you have the PPE equipment and everything in place at the time that you saw this patient?
Yes, I did have the PPE equipment available, and I did use it when I went in. At that time, he was not diagnosed with COVID, of course, although we did send the test, but I did take the appropriate precautions when I did see him.
Can you tell us a little bit what happened to you after that?
About April 5th or 6th, on that Monday, I was a little bit fatigued. Normally, I have very long working hours, about 12 to 14 hours. But recently had actually been a little bit more with the COVID crisis. So that Monday, I was a little bit fatigued. I was in the office, and after doing the morning office, I just told my office staff that I'm going to take a one hour nap, which is very unusual. But I didn't really think much of it at that time because I was working hard and wasn't getting enough sleep. But come Wednesday, it became a little bit became much more severe. I was very tired, no appetite. So I stayed home. I called my office manager and asked her to just cancel the office. And from Wednesday, Thursday, and Friday, I had to cancel my office all 3 days, and actually was not even able to go to the hospital. I stayed at home. I was sleeping 16, 18, 20 hours a day, not eating anything, just very poor appetite and just very, very weak. I unfortunately did not go to the hospital earlier as I should have. I did talk to a couple of my physician friends. One of them was an infectious disease consultant, Dr. Nayak, as well as with Dr. Khan and Dr. Pengson, and they all advised me to go to the emergency room. But as physicians, we don't do the smartest things for ourselves, even though we advise smart things. So I didn't go. Now come Saturday on April 11th, I was much worse. Now, if I walk for 10 to 20 feet, I would be very fatigued. I didn't really appreciate any shortness of breath, however, but just very weak. So at that time, I said, okay, it's time to go to the hospital. So my family drove me over to Parkview Community Hospital, which is the hospital where I work in Riverside. When I went there, my oxygen saturation was only 85%. My blood pressure was low. So they obviously took me in right away. I did a CAT scan of my chest, which showed bilateral ground glass opacity, which is consistent with the COVID-19 pneumonia. They did the COVID test, but the COVID test takes some time to get back. During this time, a couple of my other friends, Dr. Nayak, Dr. Pengson, and Dr. Khan were there, emergency room doctors were there. And then, they figured that my best chance would be if I can get this medicine called remdesivir, which at that time it was only available through clinical trials. Since then it has been released for the general public. So they called around to see where it was available. It was not available at Parkview Hospital. In fact, it was not available anywhere in the Inland Empire. It was available at Hoag Hospital in Newport. Fortunately, I have a couple of friends there, Dr. Sharma and Dr. Pandit. Both were, Dr. Sharma is a pulmonary critical care specialist, Dr. Pandit is a hematologist, oncologist. So they contacted physicians who were doing the clinical trial, Dr. Fee and Dr. Robinson, and they said, yeah, I would qualify to the clinical trial if I can get to Hoag. The hospitals, unfortunately, don't take any transfers of COVID patients. And so my daughter drove me from Parkview Hospital to Hoag Hospital, which is about a 40-minute drive. The hospital gave me oxygen. I was on 2 liters of oxygen at Parkview Hospital, which was bringing up my oxygen saturation. But on Saturday, April 11th, by the evening, my oxygen requirement had gone up to about 5 liters, just in a matter of a few hours. I did get the remdesivir that night on Saturday, April 11th. But by Sunday, April 12th, my oxygen saturation had dropped significantly, and I was requiring high flow oxygen at 60 liters and gradually titrated up to 75% FiO2. I got the remdesivir. It was on a 10-day course. the trial at that time was, so excuse me, the trial at that time was for mild to moderate cases was a 5-day course with a control arm. But for severe cases, which I fell into, it was a 10-day course and did not have a control arm. So I was sure to get the medication. Over the next 3 to 4 days, my condition continued to deteriorate. On that Tuesday, it was the condition was the worst. I was on high flow oxygen at that time, requiring high FiO2. My respiratory rate was in the 40s. I was using all my accessory muscles. Saturation was running anywhere between 88 to 90%. So it was quite worrisome. And I knew what was coming up next, which was intubation. And the survival from intubation is very low with COVID-19 pneumonia. So that night was very, very emotional, was very difficult. I talked with my family by video conference and said my goodbyes. Which was very difficult, talking to the children, talking to wife, mother, sister, father, everyone. It was an experience that I hope no one has to go through. During the night, they gave me some Lasix for diuresis, proned me again. And that night I really wasn't expecting to wake up in the morning on my own. I was fairly sure that I would get intubated because I couldn't, even turning in bed, my saturation was dropping, even on the high FiO2. However, during that night, I did prone, which was difficult to do, especially when you're using your abdominal muscles to try to breathe. By next morning, I was a little, not better, but I was not any worse than the night before, than the last night. During this time, Dr. Sharma, who's a pulmonary critical care physician, also has been my friend for 30 plus years. We were in fellowship together. He provided a lot of emotional support at that time. When I was there, I told him that, I'm not going to make it. He knew what I was talking about. And, he had the same intuition that I was not going to make it. And we're very close friends. He had tears in his eyes and, he just told me that I have to fight. I have no choice, but I have to fight. And, his words were that you cannot give up on me. So at that time, emotionally, I regrouped a little bit. And I did hang on to the night. By the morning, as I said, I wasn't any worse, which gave me some encouragement. As a pulmonary critical care physician myself, I know what these parameters mean. If you don't continue to deteriorate, that's a very positive sign in certain cases, and mine was one of those cases. I was still requiring anywhere between 75 to 100% FiO2 on 60 liters high flow. During the day, continued with that, continued to struggle to breathe. Saturation was still running in the high 80s and low 90s. Again, every night at 11 p.m., I would get the remdesivir. Over the next couple of days after that, I started noticing some improvement. Very mild. First 2 to 3 days, I had no appetite. Breathing, of course, was a problem. Another physician there, Dr. Pandit, was bringing home-cooked food for me, Indian food, because that's what I enjoy. The first day that she brought it, there was salad, and even by chewing the tomato, my saturation dropped into the 80s. So next day, she brought pureed food. Excuse me. When I started to get better, that was very, very encouraging. Over the next day, my appetite came back. My oxygen requirement went from, they had to come down to 75% and 70% and 60%. Still on the high flow. But over the next day or so, I was able to get off the high flow and get onto nasal cannula oxygen. I was discharged on Monday, April 20th. And for 2.5 days before that, I was on room air. But I would get short of breath by walking 5 to 10 feet of it. And it was a very small room, so just 5 to 10 feet, I would get short of breath. But I was able to at least walk that. A few days before that, I could not even walk the 5 to 10 feet. the infectious disease doctor, Dr. Robinson, came in every day. All the doctors came in every day, and he told me initially that I would get worse before I started getting better. So I knew the crucial thing was to really stay alive during the phase when I was going to get worse. If that could happen, and if I could stay off of the ventilator, then I had some chances of pulling through. But as I was getting better, I am also aware of other complications that can happen, such as pulmonary embolism and stroke. During that time, we had increased risk with the COVID-19 infection. So that was always in the back of the mind, but I tried to block those thoughts when they came to me. I just concentrated that, okay, I'm getting better, and that's it, I'm going to get better. And then on Monday, April 20th, all the doctors said that you're good to go home and you have to rest. My son came and picked me up. I had a very nice reception at my home at the gate. My mother was in isolation because of the COVID-19. My wife, 2 children, my brother-in-law, Tony, and my niece, McKenna, and sister Seema, they were all there, had banners for me, and it was as if I came back from a war and survived it. And since then, I was home for a couple of weeks. Initially, when I came home, if I climbed up 3 steps, I would have to stop. My oxygen saturation would drop into the mid-80s. But now I'm able to climb up the flight of stairs at a little bit slower than normal pace. Oxygen does drop a little bit, but I'm able to do it, and I do breathe a little bit faster by climbing up the flight of stairs. Last week, I started work a little bit, started seeing some patients at a very light load, probably working about 4 hours, 5 hours a day. The rest of the time, I'm just working at a much slower pace than I normally do.
It's a horrifying experience. It might be even worse because you knew exactly what's going on every step on the way. Are there times that you thought, I wish I didn't know anything what's going on?
In certain ways, the knowledge of medicine, in some ways, I think it worked against me. If I was not in the medical field, on the day that I was sick at home and was very fatigued, my friend's physicians were advising me to go to the emergency room, I probably would have just gone to the emergency room, and that's what I would have told my patient if the patient called me with those symptoms. But being in the medical field, I just thought, that's okay, I'll get over it. What are they going to do different in the hospital? These kind of, which in retrospect was very silly, and if I had not come through, would have been a fatal mistake. So in that respect, it hurt me. Once, however, I got to the hospital, I think the knowledge was okay to have because it gave me some insight. I was a very good patient. I followed every advice. I was not arguing or discussing or asking 1,001 questions. What if, or anything like that. But I think the knowledge, once I got to the hospital, helped me a little bit, except for, I don't know about that night when I was very critical, if having that knowledge was good or bad. I really don't know. Because if I didn't have that knowledge, maybe it's sometimes the fear of the unknown is more, is worse. But here I had this knowledge that I knew what intubation was going to be. I knew what the chances of coming off the ventilator was going to be. Unfortunately, I knew about the potential complications of hypercoagulability and pulmonary embolism and stroke and the chances that they were there. But I think having the knowledge once I got to the hospital was probably okay to have. This would be my gut feeling, but I don't know the true answer to that.
So that moment when we had to say goodbye to family, they were not present, right? It was from this.
No, it was all by video conferencing. There was no visitors allowed in the hospital at all. So, and that was also very, very difficult. Though I think having them there would have been very difficult also. Saying goodbye to them in person. I just don't know the answer to that because this is the first time that I've had to go through something like this.
You have lots of people to thank for your recovery, right? But especially Dr. Sharma and the medicine called Remdesivir that is now in the news every time. And also this crew in the hospital and everyone that took care of you, right?
Yes,
Those are
a lot
heroes.
absolutely, and prayers, of course. I have a huge friend circle. I think I've been getting a lot of phone calls, texts, emails. Even now, I'm getting them from people that I've known remotely. Some persons that I've worked 10 years before they've called me. So all these things help and give support.
So maybe one of the lessons that we've learned here is that don't do as the doctors do. Do as the doctors say. Get the assistance as soon as possible, right?
Very, very important. And that really is the message that I want to get across to healthcare workers. And you said it very nicely. Don't do as the doctor does, do as the doctor says. If I would have just said, okay, what would I have told my patient at that time if he called me with this? Then that's what I should be doing. Because we give the best advice that we can, and we provide the best care that we can.
Thank you very much for sharing your story with us. Is there anything else that you'd like to add?
They're just that healthcare workers, general public, they should take this disease very seriously. For the general public, they should follow the recommendations that are coming out, official recommendations in terms of social distancing, taking appropriate precautions. For the healthcare workers, just really be very, very careful. Unfortunately, we are on the front lines, and just take the precautions. Don't take any shortcuts in terms of PPEs and gowning up and going in there.
Thank you very much once more. Thank you.
Thank you.
All right. So for all medical doctors there, this happened to you guys as well. I think it's a good lesson to hear there. Dr. Malhotra, before you go, you start with a very important point when you added the issue of malaria. That's a very devastating disease that kills so many people. And by using a medicine that was not effective, overusing that depleted the supply. That's a very serious side effect, let's say, of this misinformation, isn't it?
Yeah, I think it is. So it's one of these things that it's a bit hard to quantify, but there's lots of different plasmodial diseases. Most well known is malaria, and it kills, as I said, about 500,000 people per year. There's some chloroquine resistance, but still there's a fair amount of chloroquine, hydroxychloroquine used around the world. And even if you're not from a developing country, all of us use those sorts of medicines when we travel overseas. And so the shortage is a big issue. When the government and others started advocating for chloroquine, hydroxychloroquine, the world's supplies were depleted. Our own federal government started threatening some developing countries to release their stores and threatened retaliation, other things. And I'll leave the politics aside, but I will say that the supplies of chloroquine, hydroxychloroquine are depleted in many places. And so I fear that many people suffer from the off-target effects of COVID, perhaps more than COVID itself. That's, time will tell whether chloroquine, hydroxychloroquine actually does anything. So far, the data are not very compelling that it's effective. But as I mentioned, there are more than 90 ongoing randomized trials, so we may learn more in the future. So far, from what I've seen, it may do more harm than good.
So you have very, very nice introductory remarks when you say, well, this is dangerous, a terrible disease, but this is not the end of the world, not going to wipe us out of the earth. So let's be cautious, but not be completely devastated by the disease. Do you want to elaborate a little bit more on that before we go?
Sure. So, there's some questions in the chat box about how deadly is this disease, and I'm not trivializing it at all. When you look from the Princess cruise, the boat that was there, where people were systematically tested, the mortality rates they're slightly higher than influenza, but not that much more. So influenza is about 0.1, 0.2%. COVID seems to be a little bit higher than that. We're not testing the asymptomatic cases right now. So several people asked in the chat box, how do we know the mortality rate if we're not testing everybody? And I completely agree with that. I think there are very high mortalities that are being reported because if you just test the sickest people, then there'll be a high mortality rate. If we test everybody, including asymptomatic people, I think there'll be a small increase. Not to trivialize it, obviously there's a lot of people being affected. The emotional story we just heard from Dr. Maheshwar was obviously very engaging and very compelling. And so we don't trivialize any of those cases. Obviously, it's a miracle that he survived and a testament to great care by the nurses and doctors and respiratory therapists, others involved. It's easy to sort of ascribe the recovery that we saw to remdesivir, but it's also possible that just good critical care was what the recipe underlying his recovery. Suffice to say, I'm glad he's there, and there's lots of reasons for it, but you know, people do recover from this illness. The vast majority of people are asymptomatic. I think we just need to stay calm and do what we can to minimize transmission.
Thank you very much for making the time to be with us today and share your thoughts, the professional advice. Dean Brasher, so this was a good lesson for everyone that Dr. Mashiwari shared with us today.
Absolutely. It's a lesson for all of us to be good patients as well as good doctors, and to do what we tell our patients. It's also a powerful story for future doctors, our students, to listen to, because they need to understand that this is a serious disease, and they need to understand that they too are patients. Being a physician is an incredible privilege, but physicians are also patients, and this was a very powerful story, and I very much thank you for bringing it to us.
Thank you. Dr. Bill Eisenberg, your advice to your colleagues: you are a safety officer, so that's right there on your lead, right?
Yes, well, I think, we've covered all of the highlights. Keep the social distancing. We in California have enjoyed the very low rates because we, our local leadership as well as our governor imposed stay-at-home tactics early, and I think we have benefited from that. We also have benefited from having some background good health relative to other parts of the country. And largely that is due to the fact that we do a lot of preventive care. And so it reinforces the idea that these are the times to make sure you get back in, have your hypertension, have your diabetes, get your childhood vaccinations. Those are important things that keep our background level of health high, which then serve us very well when things like COVID-19 come around.
Thank you so very much for making the time to be with us today. I know that you all have a very busy schedule, and share accurate information directly from the professionals, from the expert is the best thing to do that we can do for our community in these difficult times. Is there anything else that anyone else want to add before the closing? Dr. Mavericks, is there other? Yeah, Dr. Atul Malhotra, it seems that he has your hand physically. Yes, go ahead.
Oh, I was just waving, but I will make a closing comment if I may, and that, good critical care is here to stay, but also randomized trials are here to stay. So there was a comment made earlier about getting placebo. Once we have established therapies, and once remdesivir has been through peer review and all that, the studies are comparing to active drugs. I don't want anybody to think participating in a clinical trial is somehow harmful or you're getting substandard care. Many cases, they get very good care, and I just like to emphasize the importance of randomized trials really to make any progress whatsoever. But we're not putting patients at undue risk. We're giving them very good care.
Absolutely, and I couldn't agree more with you. The key to this is going to be trials, and absolutely, once approved therapies out there, everybody gets the approved therapy, and compared to the new therapy, there is the research is going to be the key to this, and absolutely people should embrace research, and that's where you get that at the academic health systems and large hospital systems that have those facilities. So important. It's going to be the future.
Very good comments and very good clarification at the end regarding placebo. Dr. Mavericks, are there any final comments that you want to make before the closing?
I don't want to drag it all out. No final comments. But.
All right. Thank you very much. Thank you all for being here today. Again, using the mask properly, use your protection, do your best, and let's together fight the disease. This is not the right way to do. The nose is exposed in here. This is not the right way to do. I would like to thank you all for being here, for participation, for sharing your time, very precious time. And I would like today to thank on behalf of all the organizers and all the participants, thank all the healthcare workers for the work that they are doing, putting in risk of their own lives to protect all of us in these difficult times. Finally, I wanted to dedicate this presentation to our friend Kat Sachs, that for a non-COVID related issue is not able to attend the meeting in vivo today. She was very much looking forward to attend this conversation and this symposium, but we are recording this and hopefully she's going to be able to watch that sometime very soon. We are praying for that.